Greetings, everyone. Thanks for joining us for the second of our two-day, two half-day adventures with astrovirology. We were very pleased with the participation yesterday. We had in total more than 100 people join us throughout the 4-hour event. And so no doubt today we'll have people coming on as they get out of lunch and meetings and teaching and all those things that people do. We gave a somewhat lengthier introduction yesterday, but I'm going to truncate it and make it very short today because we want to get on to our speakers. This workshop is sponsored by the NASA Astrobiology Institute in our last few months of existence, and astrovirology is something that requires a great deal more development within the astrobiology community, and we think that it's a very valuable direction to go down. And so there are two things planned for this this group as it goes forward into the future, and one is a journal article that is essentially building on the article that Ken Steadman and colleagues published, I think it was last year, and see if we can, pull this community, which is certainly larger than I feared, and making me very hopeful for the future of this, into a journal article for submission as soon as reasonably possible to the journal Astrobiology. We've been in contact with Sherry Cady, the executive editor of that, in order to facilitate that, and so she knows that this will be coming. And then the ultimate idea, of course, is to distill the important portions, the high-level messages from the community on the topic of astrovirology, and to be able to put that in as a white paper for the upcoming planetary decadal process conducted by the National Academy of Sciences. Our latest intel on that is that solicitation for those papers will begin in approximately the February timeframe. We don't know when, and so that is still a bit nebulous, but that is what we're aiming for. And then, of course, those papers will be read and adjudicated by the panels that are constituted as part of that National Academy's activity, and sometime in the following year it will come out as the Decadal Survey of Planetary Science, and that has significant consequences for what NASA is willing to fund in terms of basic science, in terms of missions, in terms of how all of these topics and technologies fit in with the NASA goals and activities. So with that, I will go ahead and turn that over to Ken Steadman, I guess. He's going to run it, and go for it, Ken.
Okay, thanks again, Penny, and thanks to everybody, both at NII Central and all the rest of you from throughout the world, as far as we can tell. I'm not sure how many of you know the reasoning behind splitting this into 2 half-day sessions. Part of the idea was to be able to accommodate as many people in as many different time zones as possible, and so that was one of the big impetus for splitting it up in terms of 2 half-days. But given yesterday, I'm really happy that I had about a day and a half to digest some of the stuff that we went over yesterday. It was awesome. It was great. We had huge amounts of information, and I will try to mention a little bit of that at the very end and try and collect a few pieces as we move along through the rest of this. I wanted to emphasize what Penny mentioned about a paper and coming together for a white paper later on. I know Gary sent out it was a chat or a tweet or something, one of these things, in terms of people who are interested, please get in touch with Gary or I or both, and we will do a follow-up to this meeting probably early next week in terms of trying to organize exactly who, what, where, when, etc., as far as papers are concerned for the next step. I think everybody has noticed that there's also a chat window that seems to be very active. We will collect all of those messages and post them to the NAI website after this whole meeting is done. Maybe tomorrow, but probably again early next week. The same thing is true for the recordings of the presentations and the actual visuals that all of the presenters have shared with us. I know there are a couple of things that people are not ready to share, which is totally fine. We've just asked that those people send those to us, and then we will do our best to go ahead and get those posted. One other request, and that is for people to ask lots of questions afterwards. Each of the talks, the first talk we're going to have from Evelyn Andreessens is actually set up to be a 30-minute talk, other than the 20-minute talks that we have later. Also with 10 minutes for questions. And again, as I mentioned yesterday, particularly for the people who are new today, if we have too many questions, you will get cut off so that we can stay on time. Those can always continue offline or in the chat box. Or if we've got some extra time, that gives us another little break so we can go and refill our coffee or have a physiology break or whatever happens to be necessary at that particular time. Just again, quick comment as far as yesterday is concerned. I thought that it went extremely well. Thanks again to the people at NAI Central, particularly Marco, for making sure that everything worked. And again, I was very pleasantly surprised that it worked as well as it did. If people have questions, particularly the presenters, in terms of moving things through, advancing your slides, etc., the key is the little share button down at the bottom of your Zoom screen, and then you should be able to share your screen with everyone. If you don't want to see yourself while you're talking, because we all just love to look at ourselves, there's also a button which you can press to say to mute your own video. So I know some people appreciate that. That's all that I had for this section. We're mostly going to be talking about virus ecology, exobiology. The three organizers are going to get to regale you with what they're doing, and I'll try and wrap all of it up at the very end. Do we have any more questions for anyone at this point, particularly for any of the people who weren't here yesterday? No? We're all good? Gary and I just got our presentations in today, so we told everyone to get them in earlier, and we managed to be the latest ones. I think Catherine was way better than the rest of us.
Bad children.
No?
Catherine who?
Hers just got sent in.
I guess that's the prerogative of being an organizer, right? You can break your own rules. We're not supposed to do that, right? And I wasn't supposed to admit it. You see, that's what happens when you let me do the introductions. Okay, so well, just real quickly again. So our speakers this afternoon, California time, and Oregon time, Evelyn Andreessens, followed by Nigel Goldenfeld, followed by Simon Roux, followed by Gary, the guy in the headless fade shirt there.
We switched our times.
Oh, you switched your times? Okay. So
yeah, so Kathy's going to go.
Kathy first, then Gary, and then I will try and wrap things up and talk a little bit about what we're doing, but mostly about sort of plans for next stages. I'm also going to try and give a little bit of background, some of the things I noticed from the chats. Not everybody is on the same page. Some of us have been doing viruses for multiple decades, some of us for yesterday morning. So I'll try to give a little bit of an introduction in terms of some of the things we're thinking about from a virus point of view. And also as a quick reminder, again, before we get Evelyn started, is that there are some great references, thanks Gary, I think, or was it Kathy, for putting it together on the website for a number of the articles, which are really nice background, with the exception, of course, of mine, which you can then go and get some more details on what's going on there. And if people have trouble accessing any of those, I'm happy to share that article in particular, and I'm sure other people have access to them as well. So without further ado, Evelyn, if you're ready to go, I think that Marco will be able to switch you over and we should be good to go.
Okay. I'm gonna try and share my screen here. And all right. So thank you very much for inviting me to speak here about virus taxonomy. And I hope I will do it justice because I've seen that there's some other virus taxonomists in the audience. And basically, I'll try to tell you what is virus taxonomy and why should you care. Feel free to tweet me. And I wanted to quickly share our beautiful new building. I work in the Quadrum Institute in Norwich in the UK, and we just moved into a new building and everybody's loving it. So just I wanted to show that to you guys. So because I'm the first speaker of the day, I thought maybe I'll just start very general and very philosophical with what is a virus. And then I'll try to explain to you what taxonomy is. And then I'll finish off with the now what. So when you look at viruses, a lot of virologists are very much specialists. And if you look at the first picture of poliovirus, a lot of people think of a virus as something small, something simple, something roundish. But then there's another human virus called Ebola virus, which is looks nothing like it, is way bigger, is stringy, ribbon-like. So the first definition of a virus, something small and round, already falls away. If you then look at bacteriophages, which are my personal favorites, and if you look at Gary's t-shirt, obviously they're also his personal favorite. In the middle is a phage that my student and I isolated during my PhD, which we call limestone. And of course I wanted to put that in because it looks like a lunar lander and we're talking about space and astrovirology. So there's loads more. I've put in an example of an archaeal virus, and it's a Lipotrix virus, and it looks pretty awesome. And it has a little hook, and it looks nothing like anything other than what you had in mind of what a virus should look like. And it doesn't end there, because the last one I have in my image is a Mimi virus, and that Mimi actually stands for microbe mimicking. And it's because this is a virus that can be looked at through a light microscope instead of an electron microscope. So a lot of people have this idea of what a virus is, and each time a new virus gets discovered, that idea kind of needs to shift. So where we're at now is that a virus is a biological entity. It goes into a cell, infects it, cells of all domains of life, and it needs it to replicate. And it's a bit of a vague description, but it's the best we can do at the time, because each time a new virus comes along, it kind of shifts our paradigm a little. But in its easiest form, a virion, the replicating free standing part of a virus, is just nucleic acid with a shell around it. And if you look at the ones on the left, which is poliovirus, they can be very simple. And if you look at the Mimi virus, again, it can get really complicated with multiple layers of capsid and these weird spikes and a port that opens, and again, it can get really complicated. So if we're looking at viruses outside of Earth, we kind of need to keep an open mind about what they can look like. I already said small is not a good definition for a virus anymore, because if you look at human viruses alone, they go from, and this is to scale amongst each other, they go from really tiny and a parvovirus to actually quite large for Ebola virus. If you were to compare them to a bacteria, that would be like this. The smallest bacteria would eclipse all of the viruses here. And if you were to compare that to a human hair, it would just look like this, because the human hair would cover everything. So not only what the viruses look like is vastly diverse, also their genetic material is very diverse. So for example, you have viruses that are made up of DNA and viruses that are made up of RNA, but not just double-stranded DNA, you have single-stranded DNA, you have double-stranded RNA, you have single-stranded RNA in a positive sense, in a negative sense, and then you even have viruses that have DNA that reverse transcribe their DNA to RNA and then to DNA again. So if we look at what a virus might look like someplace else, what we want is actually a molecule that can carry genetic information that can replicate and that's packaged in some sort of shell. One of the big questions, or one of the big questions for some people, is, are viruses alive? So people publish a paper saying that viruses should not be in the tree of life. And of course, then other people publish another paper saying, no, viruses should be in the tree of life. And there's people who are more nuanced, and there's people who say, why are you having this conversation? And I'm kind of on the train of the people who are asking, why are you having this conversation? Because it's all a matter of semantics, and does it really matter? The viruses don't care whether we think they're alive or not. For those of you who don't know about the tree of life, I'm sure all of you do, but in its simplest form, it describes bacteria, archaea, and eukarya as the 3 domains of life. And you can see that people and animals and fungi and plants are actually quite close together on that tree. If you expand it to look at all the potential types of bacteria and types of archaea, you can see that the eukaryotes kind of get dwarfed by the diversity of unicellular life. If you look at even further and you try to add all the viruses on this tree of life, you can see that the virus diversity actually even eclipses that of all cellular diversity, meaning that when you do taxonomy, stuff gets really complicated really fast. So when I say, what about taxonomy, I might want to start with, what is taxonomy? And as a good researcher, the first thing you do is you go to Wikipedia. And Wikipedia tells you not to confuse it with taxidermy. So anybody of you are confused and are actually at the wrong place, please leave the conversation now. And we'll go on with taxonomy, which is the practice and science of classification of things or concepts, including the principles that underlie such concepts. Now, I always like to share some funny quotes about that, because a person called Cowan in the 70s told us that taxonomy is written by taxonomists for taxonomists, and in this form, the subject is so dull that few, if any, non-taxonomists are tempted to read it. And it's the most subjective branch of any biological discipline, and in many ways more of an art than a science. And us taxonomists like to think of ourselves as artists, able to perceive form, shape, color, and relationships that are hidden from the gaze of the more mundane scientists. This does not necessarily reflect my own opinion. But when it comes to taxonomy, in a lot of cases, people agree to disagree. And recently I found this paper, to unite taxonomy, in the first line struck my attention, because it asks people, what do you think when you think of taxonomy? Is this an 18th century gentleman in breeches? Well, I can tell you I'm not an 18th century gentleman in breeches. And how did I get involved in taxonomy then? Well, it all started in 2011 when I was attending the Evergreen Phage Biology meeting in Olympia, Washington, and took this nice picture, or had this nice picture taken of me on climbing Mount Rainier. And what happened there is there was a couple of us scientists who just isolated a new phage, and we all realized that it was very much the same. So we got together and wrote a paper suggesting a new genus of phages called Viuna-like virus. And that was not the end of it, because writing a paper is not enough to make taxonomy official, as I soon found out. So what did I do then? I went to look at what body is in charge of taxonomy. So, and that is the International Committee on Taxonomy of Viruses, of ICTV, of which I now am a part of, and it all started at that one conference. So the objectives of the ICTV are to develop an internationally agreed taxonomy for viruses, develop internationally agreed names for virus taxa, to communicate the decisions, and to maintain an index of the agreed names. So if you want to know more about it, you can go to the website. It's a committee, so a committee has a structure. There's an executive committee filled with all of these big shots scientists, some of which are in the audience. The main is made of subcommittees who each have an area of expertise, and these subcommittees have study groups, and each of these study groups have even more specialized areas of expertise. So what happens when you have a new virus and you want to have it officially classified, or when you discover a new group of viruses that fit in a taxon? It's a semi-convoluted process that can take quite a while, starting with identifying what type of virus it is, so what study group or subcommittee you need to go to, and write a taxonomy proposal, and then you submit it, and you might need to revise, and then it gets submitted to the subcommittee chair, and then the subcommittee chair submits it to the executive committee, and the executive committee meets once a year and discusses all the proposals, and the proposals can then be accepted or asked for revisions or rejected. And after that, all of the accepted and revised proposals are voted on by all the members of the ICTV. And once these are ratified, then the taxonomy gets updated, there's a new master species list, and this links to external databases, including the NCBI taxonomy database. So that just goes to show you that it's not as straightforward as just publishing a paper saying, hey, look, I have a new genus, or hey, look, I have a new species. A quick note on what a taxon is. So basically it's a box, and humans want to put stuff in boxes, and not all boxes represent the truth, but a lot of these boxes are meaningful and helpful to go forward, and I hope that in the next part of my presentation I can actually convince you of that. So in this case, our box strategy is hierarchical, so it's a bit like the Russian dolls. Every smaller taxon fits in the next larger one, and we go all the way from realm, which ends in the suffix viria, down to the species, mostly ends in the suffix virus, and anything in between, but not all of them are necessary. So with all of this information, you're probably thinking, why should I care? So I'm going to try to link this taxonomy to virus discovery and astrovirology. And for that, I want to take you down to Antarctica. And Antarctica and the dry valleys in Antarctica are actually a very good proxy to study what life on another planet might look like, because it's the coldest and driest place on Earth. And when the explorers first arrived there, they thought that there was nothing there. But actually, there's quite a lot of life, and a lot of it is microbial life. And where there's microbial life, there's viruses of microbes. So actually, this picture that you see is full of bacteriophages, virus infecting bacteria, but it's also full of other viruses of microbes. And one of the interesting features there are hypoliths. So when you walk into this desert-like environment, and unfortunately I've never been there, only my boss has been there, you pick up a rock, a quartz rock. It's translucent, and what you see underneath is this community, and it's a complex community of cyanobacteria and heterotrophic bacteria who manage to find a niche for themselves, protected from the sun, from UV radiation, from wind, and from any of the harsh conditions that are present there. So when we investigated these Antarctic soil viruses, and how did we do that? We did that by sequencing all of them, sequencing and mostly looking at the DNA viruses, so sequencing their DNA genomes. We saw that most of the sequence data that we got was unknown. And 5 to 15% of the data that we did find had an equivalent in the database. So when we looked at what was that known fraction in Antarctic soil, it was mostly bacteriophages. And then the first time I saw one of these plots, horrifyingly, I saw all of these unclassified Cyphroviridae, unclassified this, unclassified that, unclassified that. And I thought, oh no, I'm now on the taxonomy committee. This is now my job to fix this. So it basically comes down to two main challenges when you look at an environment like that. We have the unknowns and we have the unclassifieds. And the unknowns are actually have a really apt name because they're called microbial dark matter at the moment. And for viruses, that would then be microbial dark matter, because you can break the unknown problem into 4 quadrants. You have a protein that's known in a virus type that's known. So those are the well-known proteins. But you can have a protein of which the function is known, but that's in an unknown virus genome. So those are potentially new lineages of viruses. And then you have a known lineage of virus, so a known genome, but it has this one protein in it that's unknown, or multiple proteins that are unknown. So then you have potentially new functions. But where those two meet, and what we call the unknown unknowns, that's what microbial dark matter is. And that, unfortunately, is not something that a taxonomy committee can fix. So then that brings me to what the taxonomy committee can fix, and that's the unclassifieds. So that's what we try to do, is try to bring order to the known virus sphere. How do we do that? By clustering genomes by type, looking at their shared protein content, looking at evolutionary relationships of certain signature genes, and even looking for conserved structural traits that cannot be resolved by looking at the genome sequence by itself. So in the past couple of years, I've tried to help people who are developing new tools with my wisdom that I've gathered in the last years of taxonomy. And one of the tools that was developed recently is called Gravity. And this is what some of the output looks like. And what we try to do is we can use this for any type of virus, but the example that I'm giving you now is for bacteriophages, because bacteriophages are my main love. So we just try to distill the difference between 2 genomes into one number, a number between 0 and 1. So we looked at how different or how similar proteins are that are encoded by the genomes, and look at all of the proteins, and then look at their gene order, and build that into a number, combine those numbers into a composite generalized Descartes distance, and then have this matrix of all the genomes versus each other and how much they share. So a 0 is that all of the proteins are 100% similar and are in the same order in the genome, and then the 1 is nothing similar, and obviously if nothing similar, nothing is in the same order. And when we do that for all the known viruses, we can see patterns. And the patterns is exactly what we're looking for to create boxes. And in this case, we created a new box called Aeronautics, which was a new family. A different but similar strategy was developed by the lab of Matt Sullivan and led by Ho-Bin Yang and Ben Baldock. And this is a network-based approach. So a part of it is similar. You look at the genes that are encoded by genomes of viruses. In this case, it's bacterial and archaeal viruses. And if 2 genomes share a gene, then they can get connected in the network by an edge, so one of the lines. And the more genes 2 genomes have in common, the closer together they are in the network. So we use that again to define this family Aeronautics and its internal structure. But what is cool about this method is that you can expand it into the dark virus sphere. So what you see here is the same picture as the slide before, and everything that was in the slide before is colored in red. But now it got expanded by unknown DNA viruses from the global ocean virome dataset. And this is in total almost 17,000 virus genomes that are added. And again, we're looking for patterns. And what we see is that the majority of viruses that are in environments that are not isolated, that they're new and don't cluster or cluster very far away from the known viruses. So why do we want to look at it like this? Well, we want to create meaningful groups so that we actually can use this. So I've just highlighted 2 because I know about them. So on the left-hand side in green, you have the group that I was talking about before, which has bacteriophages that are potentially useful in combating bacterial contamination in food. So if you have unknown viruses that cluster there, unknown bacteriophages, maybe you can use them for the same application. On the other hand, on the right-hand side, you have a group of bacteriophages that are known to integrate into bacterial genomes and have the potential to carry a toxin. And in this case, a lot of these carry a Shiga toxin that can go into E. coli and cause severe diarrheal illness. So if you know that your viruses or your bacteriophages cluster here, then you have the first indication that this might be going on with these as well. So the ultimate goal is to make a picture of this, of all the viruses out there, and then color all the dots with meaningful groups and functions, which is of course not ambitious at all. And this reminded me of a figure that probably a lot of you have seen before, which is a epifluorescent microscopy picture of a drop of seawater. So all the nucleic acid here in a drop of seawater is dyed with cyber green, and all of the tiny dots are virus particles, and all of the bigger dots are bacteria. And you can see that viruses and bacteria are basically everywhere. And this obviously reminded me of what it looks like at the final frontier. And you ask, is this coincidence? Absolutely. And with that, I come to the end of my talk. And I'd like to acknowledge my two main taxonomy mentors, Andrew Kropinski and Rob Lavigne, all of the members of the ICTV, my committee and my study group, and all of my colleagues at the Quadram Institute and my funders at BDSRC. And I'd be happy to take any questions.
Does anyone have any questions before I want to ask one? All right, Evelyn, I think it might be nice to talk about this some. If we're able to get taxonomy for viruses, with microbial taxonomy, once we have a certain taxonomic group, we can start inferring things that they can do. So from taxonomy of viruses, could we basically look at a certain level and determine how big that virus is or who that virus infects or maybe a biome that it lives in?
I think that's the ultimate goal. And what we're trying to do now with these big data is looking for patterns and then see if the groups that we see have something shared. So for a lot of these, it's quite easy to say in broad terms, this is probably a bacterial virus, this is probably an archaeal virus, this is probably a human virus. And then specifically for certain groups that are well investigated, like some of the human pathogens, it will be quite straightforward to say that. But for example, for the bacteriophages, a lot of it is dark matter at the moment because you could see on the tree of life that there's so many different bacteria, and most of them, a lot of them have not been cultured yet. So we don't have any information about the viruses that infect them. So it will be very, very difficult to extract information when there's no frame of reference.
I guess I'll ask another question then. So we know that viruses don't have this universal marker gene analogous to 16S or 18S. And you already talked about trying to use shared protein content to connect viruses. But we also know that viruses have all forms of nucleic acid. So how do we try to bring together all the different types of viruses? We have double-stranded DNA, single-stranded, double-stranded RNA, single-stranded. Do you imagine one big viral tree one day, or keeping them separate to start, or just any knowledge on that?
So what a group of people at the ICTV are doing, and this is research that some groups across the world are doing, is look at the very deep evolutionary relationships. And then you kind of go and look at protein folds in the capsid. And then you can, for example, link adenoviruses with PRD1 bacteriophages, or you can link tailed bacteriophages with herpes viruses. All RNA viruses share a protein function, the RNA-dependent RNA polymerase. So you can, all of the RNA viruses you can put together in a tree, but that tree is very, very ill supported at its root. So there's some controversy there. So there are methods. For example, the gravity that was developed by Gan Ayusa-kun and Peter Simmons, they broke it up into different Baltimore groups. So they have a different plot like that for each of the Baltimore groups. I see some questions in the chat as well, but yes.
Go start with Nigel, and then we'll go to the chat.
Yeah.
I've got a naive question, if I may. So you're talking all the time as if it's obvious that if you do viral taxonomy, you're going to end up with a tree. But in fact, the only reason that happens for, say, bacteria or the three domains of life is because we're in an era where massive horizontal gene transfer has been turned off, at least according to the theory that Carl Woese and I promulgated and so on. So I would have, if you'd asked me to guess, I would have guessed that for the reasons that you said about there being no marker genes and so on, that viruses would form some kind of network where the links would be whatever quality you want to associate to them. So to what extent is the thinking that we're looking for trees really built into this into viral taxonomy?
So this is exactly why I wanted to show the network, because I think the tree is kind of the framework that we are trying to force ourselves into, because that's what makes taxonomy most easily digestible if you have a hierarchical system.
Right.
But we do see, and with bacteriophages, there's a huge group of bacteriophages that have rampant mosaicism, as it's usually called. It's a minority of bacteriophages, but it's still a significant group. So one of the things, and this is my personal opinion, is that we will, at certain levels of taxonomy, we'll have blurry lines, and we'll say we cannot resolve this at this level, but we can go to the groups higher, and we can go to the groups lower. But at these certain levels, there's too much gene exchange, and that's what is clear from those networks.
Right. I mean, I think this goes back to the debate between Carl Woese and Ernst Mayr in the 1990s, where they argued over the decade at cross purposes about what was the purpose of taxonomy. And for Mayr, it was classification, and for Woese, it was just the result of what happens if you try to determine the evolutionary history of life. And so those goals are different, and they may give you completely different structures.
Yeah. And I think maybe some people get mad at me if I say this, but for me, taxonomy is about creating useful boxes to help people for the future, not necessarily to answer all the questions about evolutionary history.
Yeah, I mean, that was Ernst Mayr's view about the purpose of taxonomy. It was classification. Yeah.
Yeah.
Thank you.
My pleasure. Okay. I see a question about fragmented genomes recovering from viromes and how this affects classification. And yes, this is something that I've struggled with myself. When you have a virome data set, and for example, you have rotavirus signatures in there, and rotavirus has 11 segments. It's a double-stranded RNA virus, and you basically what do you do if you have two of the same segments in the same sample? There's no easy way to say these two segments belong together, because there's always a possibility that in one set of the virion particles, they get packaged separately as in another set. So we're not there yet. Maybe there might be a machine learning way to look at it, that we don't know the answers ourselves and we cannot see it with our eyes, but that there's a machine out there that's smarter and will fix that for us. I don't know yet. Ken, do you have other questions?
Yeah, so there were another couple of questions from the chat that may have gotten off your screen now. So do we do taxonomy on contigs, and can we get different taxonomies for two contigs from the same phage?
Yes. Okay. So we are technically allowed to do taxonomy on contigs, but we only want to do taxonomy on contigs that we have very good evidence that they represent a nearly complete genome. So we recently published a paper on it that was led by Simon Roux. And that kind of describes the framework that we're working in with regards to what we're calling UVICs, or uncultivated virus genomes. And yes, it is entirely possible that automated programs will put two contigs that actually belong to the same genome in a different taxon. And it all depends on what program you use. If you, for example, if you use the gene sharing networks of vContact, then your two contigs of the same genome will probably come into the same cluster, because if there's a near enough reference, the two contigs will cluster together. But if you, for example, use a signature gene that is single copy, as most of them are, then obviously it will only be present in one of the contigs of the genome. So therefore, they will never come into the same taxon. So this is an issue. I hope that answered the question.
Wasn't my question. There's some interesting discussions going on in the chat as well here.
I can't follow all of it yet. So I see, did somebody ask about RNA viruses?
Yeah.
So we have a classification system for all types of viruses, and not all of the methods can be carried over, because a lot of the gene sharing network, the RNA viruses are really small and they encode a very limited number of proteins. Therefore, your resolution is not as big as for the large DNA viruses, where there's a lot of proteins where you can create beautiful networks. So usually for RNA viruses, you go down to single gene phylogenetics.
Yeah, and I think this is something you've addressed already. Another question was, can such a taxonomic tree be used to look at the core components of a hypothetical virus common ancestor, similar to Luca? What are your thoughts on that?
Yeah, so there's definitely not a core viral ancestor, because viruses, there's multiple events in the past where viruses arose, and I think some people in the chat are probably more experts on this than I am, but there's definitely a multitude of different lineages, and there's no such thing as a core viral ancestor.
Is this proven already? I thought that was still a theory.
Yeah, well, it's a good question. You can't. Can you prove Luca?
Yes.
How? So it's all in the realm of the theoretical, right? And in this case, you have viruses that are so vastly different that there's literally nothing connecting them except for that we call them a virus.
What you just said would argue that it is not yet proven that there is a common origin of viruses, but that's a different thing than to say it's proven that viruses do not have a common origin. For Luca of cells, it seems it is proven there is a Luca. If you look at things like, I see Professor Gogarten is in the chat, his papers on the early hallmark genes that existed pre-Luca, you know, it's fairly obvious that there must be Luca, but I'm not sure that it's been actually shown that there was not a single origin of viruses.
Well, equally, yeah, I'm not quite sure how you want me to respond to this, because there's multiple different origins that have been deduced from the data. And linking these different origins to one common origin, I wouldn't know how to go about that at this point with the information we have.
Jason, stop being a devil's advocate here. There's, there's no data pointing to the idea that RNA viruses came from DNA viruses or vice versa. So let's just immediately assume there's two separate common ancestors.
I'm bringing it up because if we say that the question is settled, that means there's no point in actually doing a study on it, right? So if the question isn't settled, then I think it's could be worth someone investigating. So we shouldn't shut down the line of inquiry unless there's evidence that that line should just be dead.
I'm sorry to jump in here. I'll talk a little bit about this at the end, and this actually might be a good way to sort of, push off some of these discussions. I also have a paper, which I'll put in the chat here, the most recent review by Krupovic, Dolja, and Kunin, which you may or may not agree with, but we can talk a little bit more about that a little bit later on. It was earlier this year, and again, I'll put a link. I don't think we have that in our list here of origins of viruses, but I agree with you, Jason. I think that we definitely have to think about a lot of these things in a broader sense, which is exactly why we're doing this whole workshop in the first place, is to get people talking about these kinds of things, and then hopefully convincing various funding agencies that it's something that they want to support. So that's that I think is sort of the impetus, at least that was part of my impetus in terms of trying to put this whole thing together. So great thoughts, and love to hear lots of people's input as we move forward on this.
Yeah, I think we needed Mark Krupovic for this.
Yeah, unfortunately, Mark couldn't make it. There was a miscommunication, mea culpa, mea culpa, mea culpa, in terms of doing that. Apparently, he's in the UK right now, so yeah, you might be easy to find him there. So,
well, it is getting quite late here, so I don't think I'll be able to finish the whole session.
Yes, thank you so much, by the way, for doing this at, as you say, such a late hour. It's greatly appreciated.
Oh no, it was my pleasure. If there's any more questions? Any
more questions now? Do we do a 3-minute break and let Nigel get himself set up? That sounds like a good deal. Thank you so much, Evelyn. And again, certainly for being here.
Pleasure.
So Nigel, are you more or less set up?
Yes, do I just need to push the share button?
You should be able to, and you'll have to pick which screen you want to use, and then maybe we'll give it a couple minutes just in case people are coming from one place or another to try and catch your talk.
So I get this message, host disabled participant screen sharing.
Then Marco needs to help us. This
you
is where we have 3 minutes.
You should have permission now, Nigel.
Thank you. Okay, how does that look, Ken?
It looks great. Thank you.
Good, okay. And I have all these sort of buttons and things on the top and everything. You don't see those, right? Or do you?
You do not see those, no.
Good. Okay. Is there a way I can make them go away?
Marco?
You could try dragging it away.
If I hide the video panel, how do I get it back if I click that button? I'll just leave it there. It's no big deal. Yeah. And I get to talk for 20 minutes, that's right, and then there's a few minutes of discussion?
Correct, 20 plus 10. So, and if you like, I can wave at you when you've got about 5 or 10 minutes to go.
Oh, it's okay. Yeah, sure do that. I have a timer running here.
Perfect. So if you're ready to go, take it away.
Is everybody else back from breaks?
If they aren't, that's their problem, right?
Okie doke, thank you. All right, so welcome also from England. I am going to change gears. We're going to talk about the effects of viruses on ecosystems, and essentially what I'm going to try to point out is that at least in the bacterial world, viruses tend to get a bad rap. They're regarded as predators and bad things, and generally the word virus is synonymous with something bad. And what I'm going to try to argue is that actually viruses are a fantastic example of what ends up being a sort of a multi-level selection, and that and viruses through their activity can actually generate good in an ecosystem, help it to accelerate its evolution, and generate organismal diversity, and indeed even a rich set of ecotypes. So let me just get through the acknowledgements. The beginning ones, this is work with Hongyan Shi, who is now at the Academia Sinica in Taiwan. This was we were benefited from a lot of discussions with Tim Rogers, Forrest Rohr, Penny Chisholm, and Paul Falkowski, and supported through the NAI. And so we're very grateful to their wonderful support. So let me start then with a central question for biology that really came from NASA. And this is quite an inspiring document, in my view, the NASA Astrobiology Roadmap from over 10 years or so ago. One of the objectives of that was to try to understand the co-evolution of microbial communities, and particularly to understand the metabolic and genetic interactions in microbial communities, including viruses, and how these have shaped evolution and maybe determined major geochemical processes and changes on Earth. So typically we tend to think of bacteria and viruses as representing predator and prey. Viruses being the predator, the bacteria being the prey, but I'm going to try to argue that this view is a little bit too naive and too simplistic, and that the relationships between the organisms that you see first hide a much more nuanced dynamic which you can understand. The other thing I want to talk about is the environment as well. Typically, when we talk about predator-prey dynamics, we don't really think about the environment. At least ecologists tend not to, and we're going to say a bit more about that. On the viral side, and the question is, how are viral and bacteria communities stabilized? Because if you have highly diverse microphage communities, naively and theoretically, you would expect that stability would be a huge problem because of the exponential growth of phage due to their large birth size. So it's still a problematic issue. How are such communities stabilized? What controls their diversity? And of course, one simple answer is that you have spatial heterogeneity, you have spatial variations, you have reservoir effects, and so on. And that certainly is going to tend to promote coexistence, but still, when you do simple calculations, it's hard to see how it is that microbial communities can be stable in the presence of viruses, and how does one modulate the virulence of the phage to achieve that. So the way I want to approach this is I want to start with the environmental context, and I want to go back to ask what actually is life itself. Now, I would argue, along with Everett Schock and others, that really the purpose of life is to help planets approach equilibrium. And what is the basic idea here? Well, the idea here is that as a planet is formed, it cools and huge numbers of metastable states are created due to the existence of redox gradients, biochemical gradients, and so on and so forth. And the system will only be able to approach equilibrium when that chemical energy can be released. So the idea then is that the way to think about the Earth is that it is essentially a system like shown on the right here, where you have a battery, which is the creation of the various chemical potential gradients. And then what you have across that are two pathways, an abiotic pathway and a living pathway, shown here as parallel resistors. And the idea is that the environment provides energy input that drives both of these processes, the abiotic physicochemical processes and the living systems one. But living systems have an advantage because they can use novel catalytic pathways to accelerate reaction rates and outcompete abiotic processes in order to release these free energy gradients. Now, how is it that living systems can do that, whereas abiotic systems cannot? Well, the idea is that living systems discover these pathways through the flow of information. So in other words, evolutionary mechanisms impact ecology and the global planetary environment. And what I want to talk about mostly, for most of this talk, is the special role of horizontal gene transfer as an evolutionary mechanism that is unusually effective in discovering novelty and allowing systems to equilibrate their environment, or at least to tend to equilibrate it. Now, the organism that's the star of this talk is Prochlorococcus. It's a tiny marine cyanobacteria and their phage, and I'm going to use that as an example of how life uses information flow to thrive and occupy new planetary feedback, planetary niches. Now, ecosystems can be thought of in the following way. And on one level, one has thermodynamics, which is the energy input that drives everything that goes on inside the ecosystem. But then when you drill down a bit further into ecology, you'll find that it has two aspects currently not well unified. One is population dynamics, and the other is metabolism. And right now, we don't really have a good way of unifying both of these descriptions. And then the third aspect is information flow. So what I'm going to talk about in the marine microvirus ecosystem is the environment is the photon gradient, the population dynamics is cyanobacteria and cyanophage in the predation, and the information flow is the mechanisms of horizontal gene transfer. So what I'm going to show you now is that through horizontal gene transfer, through the collective dynamics of cyanobacteria and cyanophage, one can understand the range expansion and niche stratification of a Prochlorococcus. And what we're going to see is that predation really stabilizes the ecosystem and allows it to find its niches. So again, the usual assumption then is that bacteria are prey, phage are predators, but what we're going to see is that the relationship is much more complex. There's a co-evolutionary arms race between the bacteria and the phage through the cell surface receptors, which the tail sequences of the phage lock into. And so what we're going to see is that you get a diversity of bacterial genome sequences as a result of that co-evolutionary arms race. At the same time, horizontal gene transfer between the bacteria, which is mediated by the phage, gives rise to genetic diversity. The Prochlorococcus is the world's most abundant photosynthetic organism. It has a highly streamlined genome, 1,700 genes in about 1.65 megabases. It's very dilute in the ocean. It fixes the same amount of carbon as global croplands, and very importantly, it has no real immune system. There's no CRISPR, there's no prophages, very limited restriction modification. So the genetic diversity arises from genes acquired by horizontal gene transfer, code for cell surface receptors. The diversity is huge. It has the pangenome is 84,000 genes, so 4 or 5 times bigger than the human genome, and especially there is niche stratification. So over here you can see, as a function of depth in the ocean, light intensity, this is the euphotic zone, and these two curves in blue and yellow show you the population in logarithms of two particular ecotypes, a low light adapted strain and a high light strain. There are other ecotypes here, but this coarse-grained picture is enough for what I want to talk about. And then down here you have the cyanophages, which are lytic double-stranded DNA phages, usually in T4 or T7 groups. And the most important thing about them is they carry photosynthesis genes. Now, the photosynthesis genes that they have are photosystem 2 genes, and what Matt Sullivan et al. Being the Penny Chisholm lab, discovered over 15 years or so, around about 15 years or so ago, was that these genes have been swapped back and forth over evolutionary time between the cyanobacteria and the cyanophages. And we know that from looking at the trees of these genes. The other thing about this is that the phage benefit from the photosynthesis genes. In other words, they can actually use them. And they use them because they have a higher burst size during lysis. And this has been shown by a number of studies, especially from Debbie Lindell. So what did Sullivan et al. Conclude after they uncovered this? They hypothesized that host-like genes acquired by phages undergo a period of diversification in the phage genomes and then serve as a genetic reservoir for their hosts. And then what happens is that genetic exchange across these pools leads to evolutionary change for both the host and the phage. And so the idea is you've got microphage interactions create a global reservoir of genes that benefit both the microbes and the phages. But exactly how that comes about has never been made clear, and that's what I want to explain to you next. So what we want to do is build a minimal model, not a model cursed with excessive realism, but a minimal stochastic model of the interaction between bacteria and virus. And our goal is to ask, is it possible that one can derive the existence of a collective mutually beneficial state between microbes and viruses that emerges through these antagonistic interactions of predation? And the answer is yes, and I'm going to show you how. Now, I'm going to start by giving you the basic idea, and then I'm going to rapidly go through a sequence of models very quickly, just to show you the results of calculations that we did, but I'm not going to have time to go into great detail. So I want to first of all give you the basic idea first of all. So the idea is this. We're drawing here the bacteriosphere and the virusphere. Plus and minus refer to genes that are good or bad, beneficial or deleterious. And we're not saying what the genes do for the moment, but never mind. So the idea is, first of all, that the viruses gain genes from the bacteria through horizontal gene transfer. So that's what's shown in this the first diagram here. Now, the basic point is that in the virusphere, the viruses have more mutations. And so what happens is now you have a greater diversity of these genes in the virusphere, shown here. No reason to be positive or negative, particularly just randomly mutating. And those genes can be transferred back to the bacteria, as I've shown here. Now, the bacteria, on the other hand, have a streamlined genome, and so they have a huge selection pressure only to keep the beneficial genes. And so those get enriched in the bacterial population, as shown at the bottom here. And then when the process starts again, those get horizontally transferred into the virusphere. Those genes, those enriched genes, those better genes, are now back in the virusphere. And so the viruses are benefiting from the bacteria that have filtered the good genes. Now, this is actually important because the viruses, by having rapid mutation, are prone to Muller's ratchet, and so might be expected to rapidly degrade. But the bacteria, by being a filter, provide a way of filtering out the good genes and preserving the healthy state of the virusphere photosynthesis genes. Now, I'm going to very quickly walk through a sequence of models. I don't expect you to follow every step here. The last steps are the ones that are the most important. I just want to show you how horizontal gene transfer works and how it can stabilize ecosystems. And the first model, I'm just going to not talk about photosynthesis genes at all. I'm going to talk about a mythical gene that we'll call the barrier to predation gene. And in these minimal models, what we say is a gene has a particular number associated to it. And if a bacteria and a phage meet, the entity with the bigger number is the one that survives. So, for example, if a virus with a bigger predation gene value meets a bacteria with a smaller one, then the bacteria is killed and lysed and so on. If, on the other hand, the bacterium has a bigger value of the gene, then the virus attack is ineffective. So that's the way one kind of tends to model bacteria-virus interactions. So one writes these down, and I'm not going to go through these equations in detail, as essentially like chemical reactions in between the different bacteria and the different phage. The most important thing to notice here is that these are models, these are individual level models. And what you find is that if you have just a regular Lotka-Volterra model, what happens is the bacteria will make phage, and here are the population dynamics. And what eventually happens is the phage will grow because of their burst size, and eventually they will collapse the population. So that's the first paradox. So then what happens is we consider a second model where now we allow there to be mutations, both in the bacteria and in the phage. So what I'm showing you in this bottom panel on the left here is the value of this predation gene, the barrier to predation gene. High means a good value, and this is time along the horizontal axis. And what you see here is that once you switch on mutation in both bacteria and phage, they co-evolve together and the population becomes stabilized. Both the bacteria, so the bacteria and the phage have populations that persist and they don't crash. Now, what happens, of course, though, is that the viral mutation rate is much greater than the bacterial mutation rate. And so even if you have this coevolutionary arms race, eventually the population becomes unstable, as shown here. So now what you do is switch on horizontal gene transfer. And the precise mechanism for that is not really important for these calculations, but here we used a generalized transduction. And what happens then is that by swapping the genes back and forth, that allows the bacteria to receive evolved genes from the phage, and so the population then becomes stabilized. So basically, horizontal gene transfer ensures a persistent coexisting state between bacteria and phage. Now, I skipped over a slide by accident, and this here is meant to be what happens when the genes that you're swapping are now photosynthesis genes, which can be used both by the bacteria and the phage. So once again, without horizontal gene transfer, the population is unstable, but with horizontal gene transfer, the populations get stabilized. Okay, now the problem is this: as I said before, the genes are not equally good or bad. Typically, genes mutations are neutral or deleterious, and so the probability distribution for the quality of the gene, the score, is going to be some sort of Gaussian curve, but not one centered about zero. You're going to get more deleterious mutations than good ones, and so what you would expect is that the viruses would rapidly degrade. Now, when you have range expansion, as we're going to be talking about, the demographic fluctuations are very large at the front. So these number fluctuations can be very large and lead to mutational meltdown. When you include this effect, you see very quickly that the populations would collapse. But once again, horizontal gene transfer prevents Muller's ratchet by allowing the bacteria to filter back to the viruses the genes with beneficial values. So what happens is that when you look at the phase diagram of the system, you look at the fraction of beneficial photosynthesis genes in the population as a function of the horizontal gene transfer rate, what you find is this. At small horizontal transfer rate, the transfer is not strong enough to supply good genes to beat Muller's ratchet, and the system is unstable. For intermediate values, horizontal gene transfer balances Muller's ratchet, and then for larger values of horizontal gene transfer, all the phages have got all the good genes, so further horizontal gene transfer now starts to transfer bad genes, and then the system collapses again. So what you have here is a collective state. If you turn off the horizontal gene transfer, either from bacteria to virus or virus to bacteria, what you see in this simulation is that the system very quickly degrades and is not stable. Now, the last part of this is what happens when you do this in a photic gradient. So now the birth rate of the bacteria is a function of the photon density. What we're going to look at is what happens when we start with a population of low light adapted bacteria, and we ask what happens as the system evolves in time. So let's see. So we're going to run the movie. So down here at the bottom, you see the bacteria. The red are the viruses, the yellow are the low light adapted bacteria. They're trying to evolve towards the light, but in the high light adapted region, the light intensity is deadly and the frequency of light is different from the low light adapted ones. So the bacteria are forced to mutate very rapidly in order to be able to occupy the high light adapted zone, and they do that through the viral transfer. And now what you can see is the emergence of a high-light adapted ecotype up here near the top of the ocean, and a low-light adapted one near the bottom. So what I've shown you then is that the bacteria and the phage actually help each other. You have collective behavior despite there being predation. The bacteria have a slow mutation rate compared to that of the phage. Through horizontal gene transfer, they benefit from the phage high mutation rate, but at the cost of predation. The phage have a fast mutation rate compared to the bacteria. They would deteriorate by Muller's ratchet, but they can benefit from the bacteria's low mutation rate, and they can acquire evolved photosynthesis genes that prevent the degradation of the phage. So this collective phenomenon creates a huge pangenome that leads to range expansion and niche stratification. So this minimal model explains all of these facts about Prochlorococcus in one fell swoop. It tells you the role of the highly streamlined genome. It tells you that the fact that there are no CRISPR or prophages means that the bacteria are balancing the risk of predation with the benefit they get from horizontal gene transfer. You get a huge pangenome due to the coevolutionary arms race, and niche stratification emerges from the viral-mediated utilization of the photon gradient. And the end result of that is that when you look at the cyanophage, they carry photosynthesis genes because they benefit from the improved photosynthesis genes through a larger burst size. So this is what I've told you. I don't need to go through it again. What I've shown you then, the take-home message, is that an ecosystem can evolve to occupy niches where it can utilize the energy source provided by a photon gradient, and this happens due to information flow mediated by horizontal gene transfer. So let me finish and thank my collaborator Hongyan Shi, thank my funding source, and I'll be happy to take questions.
We're going to open it up for questions. I know we have a couple in the group chat, so if no one has any burning ones, I'll repeat some from the group chat.
Go for it.
One comment was made in regards to what you were talking about for the benefits of the bacterium and the virus, saying that there's not a clear benefit of the phage that we see. It means that they have to acquire the same genes over and over and over again.
The phage are acquiring the photosynthesis genes back and forth from the bacteria. That's right. They are being transferred multiple times. There's phylogenetic evidence that this has happened in Prochlorococcus and their phages, and that's what this model would predict would happen. And by the way, the ocean system is not the only system where this could happen. This could happen in lithic communities as well. It could happen in salt, stratified bacterial communities, salt mats, and so on. So any community where you have stratification and strong selection, this mechanism could operate. Did that answer the? I didn't see who the questioner was, but did that answer the question?
We have another question that kind of ties in with it. I'm trying to figure out how to phrase this. Do you see what I'm talking about, Ken?
I'm not sure, but is it the the cyanobacteria. So well, why don't I'll do my question. I think it relates to one of these, and that has
Okay.
to do with, you mentioned that the models suggest that you have a very large pangenome in Prochlorococcus, partly because they have this, lack of CRISPR-Cas or other kinds of
Right.
defense mechanisms. Do people see the same sizes of pangenomes in systems that do have more protection? How much is known about that? I don't know. So I'm just curious.
No, I don't think they do. This is a very large pangenome by bacterial standards, and our model predicts that this is really an outcome of the fact that these tiny bacteria just don't have these defenses, so they're forced to make a living in this way. They make a virtue out of necessity.
I guess I'll follow up with a question. Can you use the model to guess which genes would be acquired for a pathway? So for photosynthesis, we typically see PSBA and PSBD for photosystem 2,
Yes.
but can you make predictions from that?
So I think you could, although we haven't tried to do that. I mean, I think the most important thing is whatever is the rule is that whatever is the strongest form of selection that is going on, those will be the genes that will be swapped back and forth between the phage. I mean, the bacterial community will, if it's going to survive, it will use this avenue to to survive. And so if you want to know what is the strongest selection pressure that the host is undergoing, look at what genes are carried by the phage and ask how did they get there, and that will be the reason. So could you have predicted this? Well, probably you could have done, for the reason that you said. We understand a bit about photosystem 2, so you would have you might have predicted that these would be the genes, because that's obviously the form of selection that's going to be important for this system. But you could do the same thing, and we've suggested this for other ecosystems. One would actually be able to predict certainly what those genes would be, I think, and certainly what the classes would be. Yeah, good point.
Nigel, I have a comment. This is Penny.
Hi, Penny.
It's not really a question. It's just an observation, and that is that the value of having this very large reservoir of information available for the photosynthesis process seems analogous to me in terms of the nitrogen fixation process,
Yes.
which is widely distributed through so many different organisms and is so fundamental. And there are many variants, of course, as we now know, of the nitrogenase NIF genes and so forth. And it seems like it's a similar circumstance with similar import to the biosphere as a whole.
Yes, I agree, and I suppose from the planetary perspective, this is a way from the planet's point of view. This is a way to ensure the greatest amount of dissipation of free energy gradients.
Yes.
So life is doing that for the planet, as it were.
And so that seems actually sort of Gaian to me.
Well, I don't know whether it's Gaian or not, but it certainly is a dynamical mechanism that that is implied by the existence of horizontal gene transfer, and the way that involves the population dynamics along with the metabolism of the organisms. It doesn't necessarily mean that a system can't go unstable, but it certainly is a way for well, I think one of the burning questions is life an inevitable consequence of the laws of physics, and is life something that is generic in a planetary environment? And the sort of argument I was presenting at the beginning, which many years ago I think I heard first from Everett Schock, that sort of argument regards life as a planetary process and as something that is a way for matter to organize as an information-bearing system in order to, not in a teleological way, but to have the effect of increasing the rate at which a planetary system approaches equilibrium.
Yeah, thank you.
Okay,
Thanks.
thanks. Thank you, Nigel. I think we need to move on to Simon. So let's thank Nigel again. And see if we can fire Simon up. It looks like this is a host, so we should be able to go.
Just need the screen sharing.
Is my screen sharing turned off?
Yours is, but the host need to re-enable participant screen sharing because before I can do anything, I guess.
Yeah, Marco should hopefully be able to take care of that.
I've made you a co-host, so you should be able to share now, Simon.
It doesn't want me to. I get the same error message that I had, host disabled participant screen sharing.
Okay, try now.
Sorry, no.
Okay, I can bring up your slides.
We can do this, yeah.
Okay.
Okay. Do I have? Yeah. Okay, I got it on my slide. Perfect. Thank you very much. It's actually a pretty good segue from what Evelyn presented earlier, and then Nigel, and then actually what was presented yesterday. What I will talk today about is mostly three main things. The first two are maybe more technical, and they are mostly, why do we even care about viruses in the environment, and how do we try to study or even find them? So that's mostly sections for people who might be interested into getting into the virus world, and especially looking to do virus analysis through metagenomics. You will have a lot of resources in there, at least some pointers, I should say, to what you can and can't do. And then the third part is about the latest very, very, very new story we just got from my postdoc, and it's about some viruses dynamics, which will also tie into what Nigel just presented and what Josh presented yesterday, and Rachel as well. So that's resistance, viruses dynamics, and everything. That's pretty cool. I'm pleasantly surprised how well this all ties in together. I don't think I need to delve too much on this, but that's my typical intro slides. Microbes are important. Microbes are everywhere. Microbes are very numerous and very abundant, and microbes are pretty much controlling everything that happens on this planet at scale, almost. If microbes are so important, of course, viruses of microbes are consequently very important. We tend to focus on these tiny bits of viruses, these pesky human viruses, but viruses of microbes are this gigantic world outside of this and completely dwarfing them. Evelyn already showed the same type of picture. If you want to have more of a visual representation of this virus world, if you were not here for her talk, this is one drop of seawater concentrated and then stained with cyber green, which will reveal DNA. Big dots like this, and I guess you can see my mouth, right?
Yeah.
Perfect. So these big dots here are microbial cells. All the small dots behind are viruses. And so you can easily just guess from here how many different viruses there is in just one drop of seawater. If you're a number person, that's the kind of numbers we are. These are not very well constrained, which is kind of a common theme across all of virus ecology. None of our numbers is extremely well constrained, but it gives you an order of magnitude, right? So that's the number of virus-like particles, so the number of things which look like a virus and are most likely a virus that we can observe in 1 milliliter of seawater, in 1 milliliter of fresh water, or in 1 gram of soil. You know, we are in the tens of millions, if not billions. So that's kind of the scale at which we are looking at, or the kind of scale we are looking at when we are talking about this virosphere. Now, what do viruses do? If they're so, they're very abundant and everything, that's cool, but what do they actually do there? Fortunately for me, most of this has already been covered just in the last talk, so that's great. But of course, microbes will influence microbial community structure because viruses kill microbes, and so microbe community will be influenced by this. That's another schematic of the same log-table dynamics that was just described. So you start from a diverse community, one species proliferates, and then the phage specifically infecting this microbe, or bacteria in this case, this green one, will kill most of these cells, and eventually this will restore or return the community to some kind of equilibrium and at least a higher diversity. That's a log-table dynamics which apply to phage and microbes is usually deemed kill the winner. Viruses also shuffle genes from one cell to the next. I actually don't have to do any introduction to this because Nigel just covered it perfectly. But just remember that viruses do shuttle genes from one microbe to the next, and that influences microbial genome evolution over large time frames. And then when viruses enter cells, they also typically do more than just replicate. They will take over the cell, and they can tweak the metabolism of these cells to their own benefit. And when many viruses infect many microbial cells, that can have very large-scale impact. So here the illustration is from one of Maya's paper on the cyanobacteria cyanophage story. So again, Nigel presented this model just before, but just to recap it, the cyanobacteria is this green cell, cyanophage is this orange virus infecting it, injecting the DNA, and in the genome of the cyanophage are a number of genes involved in the cellular metabolism that the phage acquired from the host through horizontal gene transfer. And these genes are expressed and are really used to redirect the metabolism towards what the virus wants, which is more nucleotides to do more virus progeny. And again, it's more of an indirect consequence, but if a lot of viruses are doing this at the same time on a lot of microbes, this will change the way these microbes can process nutrients and eventually just affect global geochemical cycles around the world. So at this point, we have two things. We have a lot of viruses doing, or at least having a lot of massive impact on microbial communities. So that's a good reason why we want to know more about these viruses and these virus communities. How do we do that? That's when it starts to become tricky. As Gary mentioned earlier, we don't have a universal marker gene. So if you are familiar with 16S or 18S and the amplicon wars that just revolutionized microbial ecology in the 80s, 90s, 2000, we don't have this in viruses. Viruses are also very challenging to cultivate. I don't know if anyone here has ever tried to cultivate new phages or new viruses on a new host, but it's notoriously difficult and it's time consuming and it's really hard, just plain hard. So what we need is cultivation-free approaches which require no prior knowledge on the virus we want to, or on the virus community we want to analyze. So basically, we can't have the primer-based thing. We can't have a cultivation-based approach. We need something we would just go in and try to look at everything we can. And that's pretty much what metagenomics gives you. So that's a very simplified view of the pipeline that everyone applies for doing metagenomes. So metagenomes, in a nutshell, is taking a sample, extracting DNA or RNA, depending on your favorite bugs, doing shotgun sequencing, which just is sequencing everything randomly, usually in very short bits, at least for now. And then we piece this short sequence together, and we reconstruct genomes from this shotgun sequencing. The major difference between these two lines here on top is what I call microbial metagenome, which is a typical bulk metagenomes where we don't select for anything. The bottom one is viral metagenomes or viromes. In this case, there is a first step where we try to remove all the cells and only keep virus capsids, and then we follow the same DNA extraction, et cetera, et cetera, et cetera. The idea here being, if I can remove the cells and keep only virus capsids, most of my sequencing will actually be on viruses, and most of the genomes I will reconstruct will be virus genomes. That's at least a theory. All of this end up on the same thing, which is once you have all your reconstructed genomes, the game becomes which of these genomes are virus sequences. So just to give you, again, a quick sense of scale of why I'm excited about metagenomics, and I've been for a long time, and also why this has become such a big feature in the field. This is just a graph of the number of genomes we have in NCBI, in RefSeq, for all virus species from 2004 to 2018. And that's a log scale. So, we went, and that's mostly from isolation. So by doing this painstakingly hard process of isolating new viruses, we went from little more than 1,000 genomes to almost 10,000 in, almost 15 years. And now you can compare this to the number of virus genomes we have been able to assemble from metagenomes. And it started very late. Like you can see this yellow curve really in the year 2004 all the way to 2016, we were started to understand that we could do this, but we could not do it at scale. And that's really in the last 2 to 3 years that we went from having a few hundreds of these to nearly 1 million right now. And again, it's a log scale. So this linear growth here is actually an exponential growth. We are just assembling like crazy all types of viral genomes from every type of sample. Is just becoming almost routine at this point to find new viruses in every metagenome we sequence. So that's kind of the power of metagenomics here, is just this almost boundless exploration of your virus genome diversity. Just so you know, if you want to take a look at these genomes that have been assembled from metagenomes, there is a database that we have put together at JGI, which is called IMGVR. You have the address here. You will have a bunch of information on which genomes, where does it come from, which samples was it assembled from, what we think it, et cetera, et cetera. So that's kind of the collection of these genomes and whatever we know or we think we know about this virus. Okay. So at this point, I've told you why we are interested by viruses, of course, and how we do this with viruses of microbes in the environment, which is primarily through metagenomics. And I told you, yeah, we can have tons of genomes. That's great. What's next at this point? And again, we do that since 2016, so it's almost all news at this point. So you want to have more than just a genome, and that's where we decided to sit together as a community with all these lovely folks here, and we just published 6 months ago-ish this paper in Nature Biotech in this perspective, which is a big description of what we think currently are the standard way of analyzing, describing, and reporting these UVIGs. And UVIGs here stands for uncultivated virus genomes, which is another way of saying a virus genome assembled for a virus that we don't have in culture. So primarily metagenome assembled virus genomes, although there are a few other techniques. That is why we have this more generic term UVIGs. If anyone is interested into going into this area or have metagenomes they want to look into viruses, that is like this paper specifically is a great starting point, because it covers pretty much all the range of what we can do with this data, and it tells you, okay, that's what you can do, that's the standard tool, and that's what you should be careful with when you interpret these results. That's really an amazing starting point that all of this community put together to kind of help anyone new to the field, and even the not so new. It's always good for memory refresh to just have this reference. Okay, what is in this briefly? First, we have a list of the tools which are right now available and broadly used. Pretty much that's a list of what I would argue we know how to do. So identifying virus sequences in genome or metagenomes, We are not perfect at it, but we kind of know how to do this. Having a sense of the distribution and abundance of these genomes, so basically looking at different metagenomes and telling you if the virus is there or not there, we kind of know how to do this as well. Functional annotation, we also know how to do it. We don't do it well, but it's more a question of having the reference than actually the methods for how to do it. So again, these are tools that are available, broadly used, and we'll give you the standards tool and the standards way and pipeline to do this. If you don't have the resource to do it on your own, you have actually free online platforms with these virus-dedicated tools that exist. I've listed them here. The main one right now is called iVirus. You have the docs here. It's mostly leveraging what's called Cyverse, which is a cyber infrastructure, which is a fancy name to say this is a place where you can go, upload your sequence, and analyze your data without worrying about like where the computation is going or anything. It's just, for now, it's free. You just go, put your data, analyze it, and get your results. Another flavor of Cyverse, or at least another comparable tool to Cyverse, is called Kbase. Same thing, the iVirus tools are being right now being ported to Kbase, so eventually you would have iVirus in both cases. The bottom line here is that if you want to do this analysis and you don't really know how to start or where to start, this is a great starting point in terms of actually doing anything. Now, I've told you about what we could do, or at least what we think we know how to do. What we don't really know how to do, or what we are currently trying to understand how to do, are these three critical pieces. Taxonomy classification, Evelyn already covered it, and she already presented how complicated this was, so I don't need to convince you that taxonomically classifying partial genomes from metagenomes is not a standardized and routine thing right now. The second one, what we call quality estimation, is this idea of when you get a piece of genome from a metagenome, you know it's a virus. How do you determine if it is 10% of the full genome, 50% of the full genome, maybe it's actually a complete genome? We wish there was an automatic tool to tell you, but right now it's still in development, and in development at the stage of like, we are looking and exploring some ideas that we think could work on how to do it. So that's kind of early stage development. And the last one, I will develop a little more in one or two slides, but just to give some framework already, of course, as soon as you find a new virus and a new virus genome, one of the first questions that pops to mind is, which host does it infect? And so this, there is a bunch of methods that we kind of gather under this term of in silico host prediction, which just means host prediction based on sequence analysis. So not going back to the bench, but actually from the sequence of the virus genome, can we guess or take a best guess at what host this virus infects? And we sort of can, but there is still a lot of work in this area done and to be done until we get a nice and robust, simple tool, one size fits all that will just, you can apply to your sequence and you will get an answer. Okay, so not wanting to discourage anyone, you can definitely do this. Just know that some things are pretty much advanced, some are still in development. In a perfect world, the final results of all this is you will get virus genome, one or several, and along with this, you will get a full ecological and evolutionary context. Of course, this doesn't work, or at least it can't be that simple. And I just wanted to highlight a few of the major challenges, because if you are looking for a problem, here are the main ones for us. The first one is I showed this schematic of like, you take your sample, you just extract your DNA, blah, blah, and you end up in your virus sequence as if this was a done deal. Actually, this is not that straightforward for every type of sample. And especially this first step, if you want to enrich for virus particles, we have some environments where we know how to do this very well, some where we're still figuring things out. Just as an example, Gary has a paper not much more than 6 months ago, I guess. I don't remember exactly the date, but that's 2019 paper about trying to do this kind of enrichment in virus particles from soil. And that's already very tricky because viruses stick everywhere, et cetera, et cetera. So just know that this kind of pipelines, they look good on paper, they are not necessarily very straightforward to apply to any type of sample. In the same kind of line of thought, I have this box saying viral sequence identification. I told you we kind of know how to do it. We do, but it's not perfect. And there is, I just wanted to flag this out. There is a very nice paper by the lab of Bonnie Hurwitz in Frontiers, where they showed with nice examples and kind of benchmarks what are the typical mistakes that can be made, especially when using machine learning to do this virus sequence identification. So if you're interested by this aspect, this is definitely a must check. In silico host prediction, I told you we are not really there yet, but one of the main problem we have actually is that it doesn't cover a lot of viruses. So again, even if you try to use all the tools that have been developed so far, and you really try everything under the sun that we have in our toolkit, this is kind of the results you get. So that's the same curve that I've shown earlier with the number of uncultivated virus genomes. This time it's actually linear scale, that's why it looks slightly different. But the important part is on the right. In this nearly 800,000 genomes at this point, we have a host predicted for 5% of them. So we have 95% of them for which it's not even that we don't know if this is right or wrong. We actually don't have any prediction on the host. So that's a huge area for improvement and a huge, like a critical key for the field to move forward will be to kind of figure out how to get a host prediction or host information for this 95%. And then, if you want to know more about this, again, just a few references. I forgot this. We talk about this in the Mayovic paper. Rob Edwards and Bas Dutil had a very, very nice review a few years ago about this aspect of in silico host prediction. It's still definitely relevant today. So go and check this if you're interested by this question about trying to predict a host based on a virus genome. Final set of challenge. Seeing a virus in a metagenome is not the same thing as actually knowing that this virus is active and infecting its host in this environment. There's kind of this discrepancy between you see the DNA versus you actually have an active virus. And the latter is really what you want to get at for any ecological studies. So say otherwise, if you see a virus sequence in a metagenome, it doesn't mean that you have an active virus, and we need something to go at activity. Fortunately for me, I don't need to do anything because Gary will present one type of experiments you can do to try to get at this activity level. And so I don't need to talk more about this, just stay tuned. And I guess it's not the next talk, anyone, it's the one after this. But just stay tuned until Gary's talk and you will have a nice example of what you can use to try to get at this activity level in terms of data. So that's enough technical talk and that's enough kind of generalities. Now to some biology. And what I will present here is work from my postdoc Maureen Berg on green sulfur bacteria and the viruses infecting green sulfur bacteria, of course. So a very quick background on green sulfur bacteria, which I will call GSB through the whole thing. They bloom every summer in most stratified lake, and that's what you have here. These are from a lake in the Alps, and that's GSBs detected by PCR or qPCR. And you have the month of the year, and you can see in the summer you have this nice bloom of GSBs right at a specific discrete depth. And this is another strain of GSB also blooming here. And the question we have was, which viruses infect GSB? GSB are notoriously difficult to cultivate and difficult to maintain. So right now we have absolutely 0 phage isolates for them. So we have no idea which viruses infect them, or if even one virus infects them. So we wanted to know first, are there viruses? Which are these viruses? And we were very interested, or I was very interested into which type of infections will we see if we find viruses. We will find these lytic viruses, and which lead to this arms race and very active infection. You could say yes, because GSBs are very abundant when they bloom, and so this is a kill the winner. At least on theory, this is a good ground for kill the winner, in that there will be plenty of hosts, and so you could have a lot of lytic infection going on, and a lot of, again, arms race, coevolution, everything. Alternatively, what you could have is lysogenic infection, and I think Josh really presented this already yesterday. Just to remind everyone, lysogenic meaning the virus enters the host, but then stay put for a while, divide along with the cell until it's reactivated and actually then go through its host takeover and virion progeny generation and eventually host cell killing. The reason for lysogeny, or that is the argument or the rationale for lysogeny in this case, will come from the strong seasonal variation. You have GSB very abundant in the summer, almost disappearing in the winter, and that has been shown as, or at least that has been hypothesized as a good driver for lysogeny. And if you're interested by this specific and how life history traits of hosts will correlate or not with the type of infection, you can see a very nice paper by Touchon et al. from Pasteur here for anyone interested. How do we find viruses for GSB if we can cultivate them? And how do we actually understand and get into this? Well, the thing we did is we partnered with Trina McMahon in Wisconsin. We sampled, or they sampled, Trina's lab sampled this trout bog lake. This is the same kind of plot I just showed. This time it's a heat map not based on 16S PCR, but on flow cytometry counts of GSBs. So you can see as you go from June to October, you have a very nice bloom. The white bar here is the oxygen barrier. So above there is oxygen, below there is no oxygen, and we have this bloom just right under this barrier, which is exactly what you would expect from GSB. And now what we decided to do is what we call targeted metagenomics, which is a fancy way of saying, because GSBs are green, you can use flow cytometry to separate them from the rest of the cells. And then if you separate enough of these green cells and put them in one well and do a metagenome there, any phage you find there, at least on a consistent basis across replicates, should be a phage infecting GSBs, because you have nearly 100% of cells in there being GSBs. So that's what we did, and I will cut a long story short, but basically the idea here is in silico prediction will typically tell you who could infect whom, who has the potential to infect whom, or in case of CRISPR, who used to infect whom in the more or less recent past. This will tell you which virus were in these cells at the exact moment you sampled. So that's a slightly different view on the host cell interaction that are going on in your system. Now, what did we find? Again, going pretty quickly here because the interesting part comes after. But we had two distinct host populations, so two hosts, two GSBs. These are two species, pretty closely related. And then we have historical data because we have metagenomes for the same lake since 2005. And we found two viruses consistently associated with each host. We actually found more viruses than this, and they are all novel, of course, because no one could isolate a phage on GSB before. So that's kind of regular news for virus metagenomics. Everything is novel. But what really was interesting is like these 4 viruses in total, 2 for each host, which we found in 2005 and again in 2018. So of course, when you have a time series, you want to look at what the dynamics look like. And the first one looks actually surprising to me. But I will just guide you through this plot. So these are all the different years that were sampled from 2005 to 2018. The y-axis is relative abundance on a log scale, and then you have 3 curves, one for the host in blue, and then the shades of pinks for the 2 viruses associated with this first host, which we call genome A, because we are not very original so far. And you can see that, especially after 2008, the curves are literally on each other. So it's the relative abundance of the host is exactly the same as the relative abundance of the virus, and it's just go on for more than 10 years until 2018, where you start to have a little separation at the end of 2018 with one virus becoming slightly more abundant. So that was very surprising, and then we just filed it on the side and looked at the second host, and that's where things became both interesting and actually more familiar. The second host, which is here, and actually there is a mistake here. This should not be GSB genome A, it's genome B. Yeah, the title is right, the legend is wrong, sorry. So again, this is relative abundance. The genome is in red, host genome, the virus are in green. And this time we don't have a perfect correlation between genome and host. We actually have a lot of variation. We have viruses that are here and then not observed anymore, like you can detect them in 2007, this light green, you don't really see it in 2008, even if the host is here. So you have something happening, basically. You have some dynamics, you have some changes happening, and that's what you would expect for a virus and a host on such a long time scale. Like that's 13 years. So this looked a little more familiar. So we started to think about, okay, on one side, we seem to see stable association between, and actually happy coexistence between virus and host. On the right side, this seems to be more consistent with arms race. Can we show this a little more? We can, because we can assemble genomes from all of these metagenomes and compare them across time points. And this all kept being consistent with our first hypothesis. So if you assemble genome from 2007 here, and I'm talking about the virus genome here, if you compare the genome assembled from 2007 on top to the genome assembled in 2008 here, so I'm basically taking a genome from the virus here and a genome from the virus here, they are the same. Like there is barely any SNP. Like it's 99 point something percent identical at the nucleotide level. So basically nothing changed. They just stay with the host and they are happy to stay with the host. On the other end, as you would expect, things are much more complicated on the genome B and its viruses. This is this light green virus, and you can see we assembled a genome or a variant of this genome in 2007, in 2017, 2018. We never see any of these variants coexisting. We see them replacing each other, but we see clear differences from one year to the next. You have some parts of the genome here on the 3' right side that are clearly conserved. And these are the genes that are involved in capsid and these structural proteins. These are the genes that you would expect to be conserved, at least the most conserved. But then all of these genes on the left, on the 5' end of the genome, as it is right now, you have tons of small genes, hypothetical proteins, pretty much things that we don't really know what they do. And this seems to be turned over every year, most likely through a combination. So you end up with this weird situation where you have two hosts, two species of GSB. We looked at the genomes. They looked, having the same metabolic profiles, same capacity to do anoxygenic photosynthesis and everything. So they are, really, really close and similar hosts. They are under the exact same environmental constraints. They are in the same lake for the last 15 years. And they have two drastically different infection types. So the last pretty much 9 months, we tried to get at this question of why would they have different infection types? And we ended up on one hypothesis, which is microdiversity of the host. And we think this is something that has been understudied in this field of lytic versus lysogenic cycles. This is my last graph. It's a little complicated, so let me just walk you through it and bear with me. X-axis is so coverage. So in this case, it's abundance, right? So if you're on the left, your genome is not really abundant. On the right, it's highly abundant. Y-axis is nucleotide diversity. So it's a sense of how many different populations or how many different strains do you have within your population. Another way of saying this is how many SNPs do you have, how many different alleles do you have per SNPs, and how do you have a dominant allele, or do you have pretty much evenly distributed allele across your strains? For each genome, and I'm talking about the host genome here, there is 1 point for each year, and then you have this bar that just represents the spread during the year of each value. So for example, here for genome A in blue in 2007, the coverage ranged from here to here, and the diversity ranged from here to here. The reason why I'm presenting abundance versus diversity is because you expect a correlation between them, and that's just a method thing. It's like, if you don't have a lot of reads for your population, you just can't see any rare variant. So as you increase your coverage, and as you have more reads, and you sequence more deeply into your population, you should see more and more nucleotide diversity. And that's pretty much what we see for genome B. Again, remember, genome B is this genome where we have this expected arms race dynamic with the virus. We have cases where it's low coverage, low diversity, and the 2 years where we actually have a lot of diversity, a lot of coverage, we actually see a good amount of diversity as expected. Genome A is different because genome A has this weird, very, very high coverage, low diversity region here. And these are the years where we found the exact correlation between virus and host abundance, 2008, 9, 12, 13. And this is actually something that has been reported before by a paper by Matthew Bendel from JGI, when they were looking only at the time series metagenomes and the microbial fraction. And what they call this was a genome-wide sweep, which is another way of saying this population, this genome A, become clonal from 2008 to 2013. No SNP anymore. It's like a single, very, very low diversity population. And that's where we started to say, well, that may be a key drivers of why we see latent infections and this lysogenic, long-term, stable coexistence with viruses. And so my model will go something like this. This is a picture we always think of when we think of virus host, right? Arms race, super quick dynamics. In slightly more scientific terms, what we see is diverse host population We think arms race because we think there is variation in phage susceptibility between population members. So some population members are more or less susceptible, which means you can have some resistance arising. And then if you have resistance arising, the resistant strains will develop and propagate and then and proliferate. And then the phage will likely have a counter resistance, et cetera, et cetera. So you select for new phage variants. And that's what gives you the kill the winner dynamic and arms race and everything. And that's dynamics associated with lytic phages or short latency. And that's what we see in genome B, which has a diverse host population. But it seems like things can also look something like this. And again, to my surprise, that's really not what I expected. But it seems like if you have a low diversity host population with most of your population members having the same susceptibility to the phage, it's way more likely that the phage is able to infect every member of your population before any mutant can arise, at least a strong resistant mutant can arise. And that's also tied into what Britt was talking about yesterday in this possibility of mutant arising or resistance arising in nature compared to in test tube. It seems to be more complicated for this kind of resistance to actually occur in nature and in our case in Planta. And that's what we see here. Like there is no resistance, and it seems like the phages infect every member of the community. And at this point, this kind of situation will be selecting for temperate and long latency period phages. Pretty much a phage infecting 100% of its host in a given environment has won the game, and it's, doesn't need to do anything more. So we have some model working in progress. The idea would be like, is this even plausible? And can we show the switch between selection for lytic versus selection for lysogenic, just based on host population diversity? But there is a key message I want to kind of convey here is this feature, host population genetic diversity, has not really been taken into consideration into lytic versus lysogenic decision, and I think it should be because it might be a critical driver of virus host dynamics. And with this, I'm sorry, I'm slightly over time. Just a very broad conclusion. We can use metagenomic for virus discovery. We have a framework for this now. Again, we have standard paper that just came out. Go and read this if you want to start in this field. Host interactions are key to understand viruses. Host linkage is a major challenge. And just remember that virus-host interactions come in way more flavor than just a virus enter, kill the cell, and then go out. Finally, just want to make everyone as excited as I am about the virus world, but basically we have so many discoveries to be made in all of these unknown viruses. I've shown you this 95% of viruses for which we have no host ID, but we have very little idea about their genome itself in terms of gene content. We have tons of genes without any function that are still highly conserved, so we think they are important. So yeah, if you're looking for a question or if you're looking for an open field or a black box, this is pretty much it. If you want to know more or to do more, a few advertisement / plugging in what we are doing next. If you want to know more about viruses and hear from these very, very great speakers on the right, you can join us in Oakland in 2020. We have this bigger symposium March 22 and March 23. Registration will open soon, so stay tuned. I will probably retweet this heavily on Twitter and everything. So just know that this bigger symposium is coming, and this should be pretty good. If you want to learn how to do the bioinformatics yourself, we are hosting what we call MGM workshops, microbial genome and microbiomes. There is a small portion about this about viruses, so you would also learn how to do virus analysis from metagenomes. These are very cool workshops. We have them every 6 months. So if you want some bioinformatics training in microbiomes, just think about this and keep this address in mind. And lately, if you have very, very cool samples and you actually want someone to sequence metagenomes for you, that's what JGI is here for. So we have our next call is for September 26. I won't go into any detail, but if anyone has some samples they want to look into viruses or they want even to look at microbes, just send me an email, hit me on Twitter, and I can either connect you to the right person or just tell you, what is the framework in which you can apply for this kind of grants. And with that, I just want to thank everyone, especially involved in this GSB story, which has all been led by Maureen, my postdoc, with the great help of Daniel for the flow cytometry, and the lab of Trina McMahon in Wisconsin and Don Bryant in Penn State. Thank you. Where is my chat?
Okay, I think we've got time for maybe a question, if we can do it quickly. If not, I'm sure that Simon will be more than happy to answer questions offline as well.
Definitely.
Just really quickly, Simon, if I may, did you notice anything specific about those two populations of the GSBs? Was there something about the genome of the host that you really noticed, CRISPRs, or something else that was variable or less variable?
So that's a good question. We didn't find any key feature that would explain the difference between the viruses. One issue we have is that they actually are so closely related. They both have a CRISPR array, and these CRISPR arrays have the exact same repeat. If you do a metagenome assembly, you can't tell if the spacer linked to a repeat comes from genome A or genome B, basically.
Got it.
That's how closely related they are. So at this point, I mean, we looked into these genomes, and we couldn't find any good explanation until we looked into this population diversity.
Cool, thank you. Okay, so Catherine, you're up next. Slight
right.
program, so it's all yours.
See if I can. Okay, so first I have to apologize. I am not Gary. I had a conflict that arose yesterday, and of course, out of all of the hours of the day, it came up that it was going to happen from 3:30 to 4, of course. So Gary was happy enough to switch with me. Thank you. So I do not work on viruses, but after this, I'm convinced that I should be. And I'm going to switch gears a little bit and talk about a framework for thinking about life detection, and then viruses as biosignatures, and go into instrumentation of how you might kind of give people, I don't know the background of how many people have actually worked on proposals for flight instruments, but kind of give the audience an idea of some of the avenues and the requirements, and then talk about the technology that I'm currently using. So if we start looking at astrobiology, it's the search for life's origins, evolution, distribution, and future in the universe. And especially after sitting in the last day or so on this, viruses, I've learned, really play a role in every single aspect of this question. And it seems so integral to this question. I'm really happy this meeting's taken place, and it's a subject that I think needs to be pushed, and we need to be looking for viruses everywhere, it sounds like. So with that, we'll need tools, and I'll get to that in the technology part. So to understand the distribution, if we go out looking for life detection, it's kind of nice to know what you're looking for. So it does pull us back to that question of what is life. And the way our theory for life right now is we're defining it based on its properties. And you can kind of think about this of how we used to define water before we really knew what water was. Before we knew it was H2O, we said it was clear, colorless, wet. We defined it based on its properties. And we do the same thing with life now. It's self-enclosed, self-sustained chemical system capable of undergoing Darwinian evolution. So we really are, when we talk about life detection, we talk about biosignatures, biomolecules, because we're looking for these properties of life. And that's why if you're here for the first day, this kind of goes back to Benner's talk about the universal signature for life. And I'll get into that in just a little bit. NASA has put out a life detection ladder. It's a nice beginning framework to think about how we might go about looking for these properties of life. If you haven't checked it out, I would recommend just familiarizing yourself with it. So there are rungs on this life detection ladder, and the top of the rung is the highest evidence for life, and that would be Darwinian evolution, right? Because that's in our definition. The problem with that is if you go to Mars or Europa, Enceladus, or Titan, observing Darwinian evolution, there we go, thanks, Ken, might be mildly difficult. And if you do look at the life detection ladder, there is, what measurements you would make for all of these proposed instruments. Another one would be growth and reproduction, metabolism, molecules and structures conferring function. I really think viruses fit into this role, and that is, if you go into molecules and structure conferring function, the polymers that support information storage and transfer, at least here on Unitarian life, is DNA and RNA, and viruses are little storage units. In my non-virus terms, I just think of them as packages of these molecules that confer function. So of course they are something. Based on this, I would think there'd be an interest in looking for. Even if, and the NASA Astrobiology Strategic Plan in 2015 did comment on weird life and recognize that alien biochemistry might not be the same chemistry that we see here on Earth. But you would still need to have a molecule that stores information, be this RNA, DNA, or something completely different than these molecules. They'll still need to be functional molecules, and even if viruses aren't as we know them, we could assume or begin to think about them still encoding for or storing whatever this information storage is. So here we have Benner for the universal feature of life. Benner, based going back to his presentation, these are the molecules encoding genetic data necessary for functioning and replication of life. And there he makes the argument for those being polyanions or polycation polymers. So that just is kind of to give you the framework about how life, if you're thinking about life detection and proposing life detection instruments or being on a life detection mission, kind of need to relate it back to sort of these benchmarks and these goals that NASA has put forward. And for life detection missions, especially for viruses, I'm sure there's a way to look at them in exoplanets, but I focus on instrumentation that would work in this solar system. I would like to see a life detection mission within the solar system before I die, or life detection mission before I die, so I focus on this solar system. With that, you could look for intact detection, which would be high resolution microscopy, and these are just some, this is not all-inclusive, this is not encompassing of all the different technologies that you could use to look for viruses. This is just me throwing some suggestions out there. So intact detection, you have microscopy, you could look for protein fluorescence, and then nanopore-based electrical sensing, and that's what the rest of the talk will be about. And then if you're wanting to break open the capsids, oil extraction, and look for functional molecules, Benner proposed an instrument in his talk, and then nano-based electrical sensing as well. So just hope that the community starts thinking about, we need to do life detection missions that include viruses, but we can't just say that. We have to start thinking about and developing instruments to do that. Because I think that is a community responsibility. If we decide that this is something we should be doing, we also have to help develop and provide the tools to have that done. So the instrument development framework can be confusing. There are all of these TRLs, which is technology readiness level. And if you just read the definition and you start trying to apply it to your technology, there comes some sort of ambiguity, and there's weird justifications that you see for different instruments being at different technology readiness levels. And this is also another reason If you are tired of seeing a lot of the same instruments go on to flight, it's because there are very few instruments that have made it through this technology readiness ladder, if you will. And just because it works great here on Earth, we know how to do it, does not translate directly into having an instrument that can do it for flight. So your technology readiness level 1 is, the basic principles, and then 2 is the concept and application formulated. 3 is when you really start getting your critical components down, but when you hit 4 and 5, that's when you start doing breadboard validation. You do an integration of all of your different components. 6, your subsystems in a relevant environment. And I should back up a little bit and say that each component, you are only at the TRL level of your weakest component. So each component within your flight instrument has to have made it to that TRL, and not just each component, but in the specific integrated schematic that you are proposing for flight. So as you can see, this gets very engineering, this gets very regulated, and it's not necessarily as biologists or virologists the ways we're used to thinking. So if you do go off to write these instrument development proposals, I would definitely recommend teaming with an engineer because they start asking questions, especially if you get higher up in the TRLs. You need to list every component down to the screws, how they've been tested, what are they made of, what are your power requirements, your weight requirements, how much does everything weigh, what materials have they been proven in flight. So it gets rather complicated. So just because we can do it here on Earth doesn't necessarily translate into an easy way to do it in space or on a different planetary body. Not to say that it can't be done, it just needs to go through this sort of hierarchical system. And there are several different proposals for doing this. There's the NIAC, which if you have a crazy concept and you want to sort of explore if it's feasible, that takes you from a TRL 1 to 2. I'm very new at this proposal. Penny is the queen of NIACs. But then there's also the Picassos and Matisses. Picasso will get you from a 1 to a 3, somewhere in there. And then 4 and above is a Matisse. So I'm sure there's other proposals. These are just the ones that I personally am familiar with. So now that we've kind of talked about instrument development, get on to, so this is what I currently am working with. I'm working with nanopore technology, and I specifically am looking for long chain charged polymers, not necessarily DNA and RNA, but anything that could represent that functional molecule. Nanopore technology, the concept is pretty simple. It can, there's much more layers of complication that be added on as you progress, but the basic concept is you have a nanopore that spans a membrane. Electrolyte solution is on either side of that membrane. A voltage is applied, an ion current established through the pore. As a particle translocates through that pore, you get a blockage for a short period of time, and that current drops. So that little current dropping right there, in the schematic, you can see you have your depth and your duration. So your depth is actually telling you the radius of the molecule going through based on the size of the pore. As it translocates, if it's closer to the size of the pore, it's blocking more current, and you're going to have a larger depth, where your translocation time is telling you like the length of your particle going through, how long it takes to go through. So the concept there is fairly simple. And right now there are two different ways of doing this nanopore technology. You have the biological nanopore. That's commercially available. It's operated, it's through Oxford Nanopore. It's operated on the ISS, which was really cool. So they did sequencing in situ on the ISS, and it worked great. Actually, with the way the flow cells work, the MinION, that's the biological one, actually worked better in the space environment than here on Earth, and not quite sure why as to yet. But for the biological one, just to explain, there's actually a biological protein that creates that nanopore in the membrane, and it's very specific to translocating DNA and RNA through cell membranes. So it is very specific to DNA and RNA. And it works great on the ISS, but when we start thinking about longer duration missions, again, one of the things that I talked about for instruments is you have to start looking at the material that you're made out of. And a biological membrane is not going to be robust enough for space flight. If you think about your temperature swings, your radiation environment, and just over time, you're going to get degradation of that protein. So it's not robust enough for a long duration mission. Also versatility. If we go back to thinking about life might not look like what it looks like here, the biological pore is very specific for DNA and RNA, so you don't have the versatility to look for other molecules of interest. Whereas with the solid state, you can change that pore size. You can have it smaller, larger. You can change the shape of the pore. You can coat the pore. You can reverse polarities, make things go backwards. So here in the picture is, if you start at the very top, there are two little electrodes going into the reservoirs. The little reservoirs are those golden things. As you can see, this is a very small, very lightweight device. It's made of silicon nitride, which has flight heritage. We fly things made out of silicon nitride all the time. So we know the material works. It's a small weight, small power requirement. And in between those reservoirs, you can see in the image on the bottom left is a little microfluidic channel. In that channel, there's a reservoir with a tiny little hole. So this is just a single hole chip. You can do multiple arrays. There are various different schematics. And you would introduce your sample into that square reservoir, which is on top of the gold reservoir is sitting on top of that. And you would wait and watch the signal as your biological molecule of interest translocates. Again, I don't work with viruses, but I thought this would be a good way to introduce how this technology is currently being used to look at viruses. So here's a paper from 2014. It looks at the stiff filamentous virus FD. You can see a translocation there, but there's also a lot of if the virus is folded, it's too large to fit through that nanopore. So you have a lot of collisions. And as you can imagine, those collisions, as the viral particles bump up against that hole, they're blocking the current for very intermittent amounts of time. So you look there on the graph and you can see those collisions happening right before the translocation. So the viral package is hitting the nanopore multiple times until it finally gets the right geometry to translocate. And then if you look at sort of that heat map, it's just showing you the grouping where you can see the collisions fall in a much broader spectrum, but the translocations cluster quite nicely. So in 2016, we did a study looking at the tobacco mosaic virus and found that there's actually a very clear pattern as the tobacco mosaic virus goes through the nanopore, because it's very rigid. It doesn't have that flexibility that we saw previously. So there are these three steps in the translocation pattern that are very, very indicative of this rigid rod-shaped structure. And you have the first part in blue there where the virus is trying to enter the nanopore, wiggling around a bit. Red where it's finally it's starting to translocate, and then green where you get the actual translocation. So as you can imagine, you could look for any virus because you can change the pore size, right? You could make it smaller or larger depending on what virus you're looking for. And once you know the indicative pattern, you might not be able to say for sure that this is tobacco mosaic virus, but you would be able to say that this is a rod-shaped virus. And you would know, you could defer from that the radius and the length of the virus. And then Wu in 2011. Doing good. So again, these are looking at the HPV capsids, T3 and T4. As you can see, their radius is 31 and 36 nanometers, right? So they're a different size, and you see that in the translocation data up there on the top. You have your baseline signal, and then your T3, which has your smaller radius. You see the translocations happening there. And again, remember, the depth of your translocation signal is how much current is being blocked. So if it's smaller, you're not going to be blocking as much current. And then your T4, which is larger, you see that jump in current blockade there, and that's because you're blocking more of the current as it translocates through that nanopore. And then last one on this, I'm just hoping to show you the ability and the diversity of the system, sort of some of the things that you can do with it. There also is a way to get at the density. So you can measure the mass of the nanoparticles or viruses and their sedimentation. So looking at the time it takes for them, you know their radius and their length, and then you know the time at which it takes them to settle. This might be different for spaceflight, but this works great here on Earth. But one of the other things that is good that this paper points out is you are working very, very small volumes and small concentrations. And this is very important to have technology that can work with small volumes, because if you're thinking about going through, say, the Enceladus plume and capturing some of the samples, so Enceladus is icy moon that is spewing sort of like a geyser, particles of water out into space. And if you wanted to fly through that and collect that, you might be getting a milliliter, I think is right now the projected volume that they're thinking of collecting. So again, you are looking for a needle in a haystack, so you have to have something that's very sensitive and can work with these low concentrations and small volumes. One more thing before I run out of time. Alexander couldn't be here, but I did want to point to a paper that was published in 2018, the need for including virus detection methods in future Mars missions. And this goes over sort of the argument or reasons why to look for viruses on Mars. And it's that we don't really know how and when viral units were created here on Earth. The virus first hypothesis is very controversial. So exploration of Mars, you could kind of finally close this gap if viral units were actually arose before cells. We can find if we find viral units on Mars but no cells, then that can only be explained by the virus first hypothesis. And if they don't exist on Mars, falsifies the viral first hypothesis. Again, this isn't my work. I just wanted to put this up here and point to the paper because unfortunately he couldn't be here with us today. And with that, I just want to say, build it and they will come. There are so few instruments that are flight ready, so get your instruments out there and work on them as much as possible and often, and you'll have a very good chance for spaceflight. And that's what I have.
I guess we'll open up for any questions anyone has.
So I'll start it off. Catherine, so as we've heard from a number of people here, particularly Evelyn right at the beginning, many bacteriophage have these sort of mixed structures, and I'll show some of the crazy archaeal virus structures in just a couple of minutes. How easy is it for these nanopores to sort of look at a head and tail phage, for instance, or something like that? It seems that might be quite a challenge.
I actually think the more crazy the structure, the better it will be at detecting it. Because as it passes through the nanopore, you're going to get a very indicative signal. So just as we saw the tobacco mosaic virus translocate, and you had that 3-step-wise translocation phase that happened almost every single time it translocates, the crazier the structure, the more specific the signature. So it actually, I think, would be a bonus versus a detriment.
Cool.
Yes.
If someone happened to have a crazy virus and wanted to put it through a nanopore, whom would one talk to?
I would say send me an email because I love putting things through the nanopore. I will take your samples.
We'll talk. We got some crazy ones.
I also just wanted to point out there the benefits we get from the long read sequencing. Not only are we getting the in situ sequencing done, but also potentially a whole virus, which is phenomenal.
Yes, the Earth-based biological pores by Oxford Nanopore, Minion and the Flongle, don't know their naming, but they're great. I've actually taken it to the Atacama and did in situ DNA and RNA sequencing there. Took it to the Arctic. It functions great in situ to kind of look at what you have kind of on the spot right there. You know, and it's commercial, off the shelf, available, and it's kind of revolutionizing the way we're able to do biology and the way that it's open to multiple different people, because the protocols for using it are so very simple. You just need to extract your DNA, and then they even will do a library prep for you on an automated Voltrax, and then the Minion, you just need to know how to pipette onto it, and it takes care of the rest. So if you can pipette or have someone pipette for you, you're good to go. All right.
It's not been done in caves.
It doesn't. Next place it needs to be done.
I got a list.
I just wanted to bring up some of the discussions going on in the chat, is that with the long read, there are some error rates.
Yes, that's very true.
So I don't know if you want to say anything about that. I have something else but go ahead.
You know, I would say that, you know, using dual methods to try to overcome the error rates is the most successful strategy that I've seen so far. So the Minion is great for taking it in the field and doing stuff in situ and for getting long reads, but then it's a good practice to always collect and process samples using traditional techniques so that you can kind of get at eliminating those errors.
That's harder on a spacecraft, though.
Right, but
Right.
for space,
testing.
for testing and for space flights, you know, depends on what you want to do, but most cases you don't need that level of resolution.
But you can definitely imagine the promise of it, because like what Arvind was talking about, segmented genomes, we have so many reads that we also throw out. Assembling viral conjugates can be difficult. So if you had a short read version and you had the long read to kind of be the reference genome and then just fix some error rates, that would be beneficial.
Well, even for looking at stuff on ISS or the lunar surface, if you wanted to see how things are responding to their environment and you don't want to necessarily send everything back to Earth, you can sequence in situ, and yes, you might have these error rates, but you'll get data in situ, which is something that we haven't really done before.
Yeah. There's another question here. Felipe, I wonder about the minimum quantity of the target sample to be able to detect the molecules in Martian soil, desert soil, etc.
So your level of detection for the solid state nanopore is really based on your level of patience. In theory, you could do single molecule detection. So if it's a charged charged particle and you are applying that voltage, it will eventually find its way through the nanopore and translocate, in theory. So the level of detection really just is based on time. In lab with samples that are, of course, we took some Lake Vida samples and it's fairly sparse. After 30 minutes, we were seeing translocations. We're not going to see everything unless we've run it for several days. But you also have, it's the smaller we're also working on a front-end sample processor for Mars or an ocean world where it takes a bulk sample, lyses, extracts the polymers, and then goes through a concentration and desalination phase, because in a lot of these environments, as you concentrate, your salts increase and you want to be working within an electrolyte buffer range, right? So you have to work on desalination, and that's one of the questions that we're trying to tackle is with this front-end sample processing system, where we're concentrating, what sort of levels of detection can we get in to with functioning within a reasonable limit of time. So that is a currently funded ongoing project.
I also want to point out there for people who maybe are intimidated by bioinformatics, because short read's been around for so long, we have all these app-based systems to help you and stuff. Now, a lot of these apps don't work for the long read, so this is an ongoing process, and there's definitely going to be a lot of increases in efficiency and workflows and stuff for bioinformatics. I think within the next 1 to 2, even beyond, just within the next year, we'll definitely see a lot more on this.
And we talk about promise, but again, this is something that is already being used at large in the community. It's not something that might be working in the future. I mean, it's used to track Ebola outbreaks in Africa. It's used, pretty much all over. There's a pilot program at the hospitals in San Francisco. If you come down with an illness and they can't diagnose it, they send it away for culturing. It comes back negative. You have to send it away again. It comes back. I think like 95% of those patients die if it's a serious disease. So now if the culture comes back negative the first go around, they go ahead and use the Menon to try and identify and get information right away. Yeah, there's I could go on and on about crime scenes and their application for blood spatter and identifying they're working in every aspect. So forensics. It is forensics. So it is progressing at a very rapid rate. And I think I'm out of time.
Yeah, I think we'll go ahead and start our break and reconvene at 3:40. No problem. Thank you all.
Everybody.
I wanted to talk about using stable isotopes generally to track viruses in soils. And I'm focusing on soils because particularly phage from soils are what I care about most. But this can be applied to any environment. And this is not the only tool that can be used to track viruses or look at the activity of viruses or microbes. So starting off that soils are complex ecosystems. In this cartoon here, you can look at about 1 centimeter squared area and see that's teeming with organisms. And this is many range sizes. We have our plants, we have our macrofauna, our mites, these nematodes, amoebas. And if we really want to look at microbes and viruses, we have to zoom in on this one soil particle. Now we can see our bacteria in some pore water. And then if we zoom in further, then we can see our viruses. So I want to point this out there because they're just here with all of these other organisms, and they can be random in random spots in the soil. And most of what we know about viruses comes from isolates, which are extremely hard to isolate, as we've heard earlier, and then from metagenomics. Because there's so many organisms with their larger genomes, when we do metagenomes of a soil environment, we get low resolution on viruses, typically less than 2% of our information goes for our viruses. From this, we've learned that there's about 10 million to 1 billion viruses per gram of soil. And because soil is structured and has microheterogeneity, the amount of viruses per host can vary from about 1 to 1,000 or even more. We've seen that soil viruses are morphologically diverse. We have our classic phage, our double-stranded DNA viruses. We have our smaller single-stranded DNA viruses. We can have some filamentous viruses and some pill-leaking viruses. But with recent metagenomics and viromics, which is separating the viral particles before sequencing, we've been able to start learning a lot. The main thing is that we've been able to recover thousands of viral populations. And these are different flavors of viruses that are similar, so we group them into these populations, and we use a 10 kb threshold to get really robust populations. By looking at their genomes, we've been able to identify that viruses can have direct impacts on microbial biogeochemistry, not only from lysing of dominant microbial hosts, but also from having the host express the auxiliary metabolic genes that they carry. In this example, we have viruses killing some dominant hosts. They have an AMG, which is called a glycohydrolase in this case. And these can help break down these complex carbohydrates, these polysaccharides, into these small monomers that are digestible and can feed in a range of metabolisms. A kind of example I like to give is imagine an environment where there's pineapples everywhere and you have to eat the rind and everything. This is really hard to do, and not everyone can do this, and people don't want to do this. So if this is broken down, and let's say you have cheeseburgers and salads around, this is great, and a lot of different people, a lot of different organisms can actually digest this and function off this. Now, I've told you from metagenomics we've learned a lot of this, and it's from these recent advancements along with these laborious viromes. But if we think back, there's another issue. Looking back at the soil, not all of the microbes here and the viruses are active at any point. So when you take a metagenome, you're just getting everything that's there, and we know there can be relic DNA in soils. So what do we actually care about? We care about the microbes that are active. And to help explain this, these are ones that are growing and actively interacting, contributing to the environment, not ones that are necessarily dormant or deceased, which when we take a metagenome, we are seeing all of this, which is biasing what we think is going on in the environment. So the goals here are to increase resolution on viruses so we can understand them and their ecology, and then target the active microbes and the viruses that are infecting them. So normally, how would you track an animal? Well, for a cat here, we have a collar. For butterfly sharks, we can put a tag on them, but viruses are way too small for this. And like I said, there's many methods, but the one that I use are stable isotopes. It's also called stable isotope probing. And stable isotopes are atoms that contain the same number of protons, but differ in the number of neutrons. So in the example I have here, we have hydrogen, which is stable and exists in the environment, and the one that we most likely see is that it has one proton and one electron, but it also exists in the form of one proton and one neutron. And the difference is we have a little bit more mass, and we can use that to our advantage. So here we can incorporate atoms, these elements, into compounds that can help us track biological processes. So here we have water, H2O, 2 hydrogens, 1 oxygen. There's 8 protons and 8 neutrons. Let's call this the Tootsie Roll. Now we can have heavy water, and this is not with deuterium. This is 2 hydrogens and 1 oxygen, but it's H2O, so it's 8 protons and 10 neutrons. So it has a higher mass. So these are our different flavors of our water. These are like our flavored Tootsie Rolls. So I'm going to give you 2 examples of how I use these in experiments. The first one is we have this heavy water and then regular abundance water, and we incubate that in soil. We can also be more specific. Here we have plant biomass, and we can grow plants with enriched carbon with 13 CO2, so that way the whole plant is enriched, and feed that to microbes too with soil. So with the heavy water, we can get at all the active organisms, because all organisms use water. And then any viruses that infect them will also get labeled. Now, for the plant biomass, this is specific organisms. We get a higher level of detail. Here, we're looking at microbes that are breaking down this plant biomass and those viruses that are infecting those specific microbes. We can extract the DNA like we would do with a normal metagenome. We can do ultracentrifugation with a cesium chloride density gradient in order to separate the light DNA and the heavy DNA. So we can have our dormant and deceased microbes in a different area than we have our active microbes that we want. So by being able to separate this DNA, we can sequence it separately, and we can do comparative bioinformatics if we want to compare that. But by separating it, we can have more sequencing power going towards the relevant microbes and viruses that we care about. And this helps reduce complexity and increase our resolution. So our goals were to increase resolution, and it's going to do that. And then we can target our active microbes and their viruses, and it's hopefully going to do that as well. So when we were conducting this experiment, our two main goals was to try it on two different types of soils that were extremely different, so different soil biomes, and then see if it actually works, if we can identify active viruses and their microbes. So the two field sites that we chose were two long-term ecological research sites. The first one was in Alaska. It's a partially thawed permafrost bog habitat, so it's frigid, it's cold. We don't expect life to be there below freezing. Our other one is in Puerto Rico. It's in a tropical rainforest in Loquillo Experimental Forest, and it's a highly dynamic tropical rainforest. So zooming in on the Alaska site, I first wanted to point out that everything you see in purple is permafrost and discontinuous permafrost, which is ground that's frozen for 2 or more consecutive years. So zooming in more at our Bonanza Creek site, where the average temperature is less than 0, so it's freezing. Sorry, we have some delay. We collected some soil cores from this field site, and I'm going to quickly go through the methods so you can get an idea of what we actually have to do. We took 2 grams of soil from the top 10 centimeters of our cores, and we incubated that, and we pressurized it to remove water and put in either our natural abundance water or a heavy water. And then we incubated that for half a year and for a full year. And from this, we collected a lot of information, but I'm only going to talk about the metagenomic information that we gathered. So looking at the 23 metagenomes, we were first able to identify and assemble 30 what's called metagenome assembled genomes. These are microbial, these are almost whole genomes that we assembled from the metagenomic data. So we found 30 microbes that are active below freezing. They're interacting with the environment below freezing. These bacterial hosts span 3 different phyla. And then so the bacteria are active. I really wanted to drive that home. This is an important concept, that even though the ground is frozen, they are interacting with their environment. From this, we were able to detect about 4,000 viruses, and we used 2 different viral detection methods. The first is Vir sorter, and the second is Deep Vir Finder. Now, a lot of these viruses can be very similar, so we grouped them into viral populations. So it's like having different flavors of the same virus or different strains of this virus. So I put up here the different methods you can go back and look at of how we group and cluster these viruses. Our next thing is to link these viruses to hosts. Now these are preliminary results. We still have more to go through, but at first we did just nucleotide identity, which we took the genome of the virus and we looked for similarity with nucleic acid within the bacterial host. So the first thing I'm going to show you is this blue heat map here. So to help you digest it, on the x-axis we have our 332 viral populations, and on the y-axis we have our different sample treatments. The 2 blue ones at the top are our natural abundance water. The 2 red on the bottom are our heavy water. And you can see we have it for a half a year and for a full year. Now when you look at the heat map, the darker the color, the more abundant that viral population is. So first, we detected a lot of viruses below freezing. This is incredible. We have active microbes and viruses below freezing temperatures. So we were able to, like I said, we could do comparative bioinformatics. We were able to separate active and non-active. So here I pulled it together for one figure. So we have our non-active at top and our active at bottom. And we have 256 active viral populations. Now, the first thing I want to show you is if we look at these viruses that were there at a half a year to a full year, we can see that a lot of viruses actually persist. 68% of these viruses that persist actually increase their abundance. And this can be for many different reasons. These could be temperate viruses propagating microbial hosts. These could be virions persisting in the soils themselves, Or this could be that they burst open, they got degraded, but the viral DNA itself is accumulating in the environment as relic DNA. If you also look to the far left, you'll see that some viruses are actually gone. So we have viral populations here that were abundant, and 6 months later, we no longer see them. We've also seen new viruses emerge. So we have a dynamic viral community that is changing just over 6 months here. So just hitting home, active viruses below freezing before I look into our other samples. So now moving on to our highly dynamic tropical rainforest. This is in Puerto Rico. I put up some maps to help reorient yourself. It's in the northeast of Puerto Rico. Oh, sorry, we got some lag going on. Okay, so you can see from here, it's a highly lush, beautiful environment, totally different than our last environment. So here we approach things a little differently. We took enriched biomass and natural abundant biomass, and we added that to 20 grams of soil, and we let that incubate. Now, we didn't want to just do that. We wanted to mimic the environment. And this environment has heavy rains that occur often. So these soils go from oxic to anoxic frequently. And how does this affect the microbial communities and the viral communities? So our 4 treatments were static oxic, and then we did a high and low frequency of oxic and anoxic, and then we did totally anoxic, which is all controlled by our headspace. So from this, we incubated for 44 days, and we generated 85 Sept fractionated metagenomes. And this is where we divided the DNA on the cesium chloride density gradient, and then we sequenced those separately. But then we also took all the DNA together and just sampled and sequenced what we call the bulk DNA. And this is a microbial metagenome, as Simon called earlier. We were able to get 95 metagenomes from this, which formed into 214 different microbial assembled genomes. So these are genomes from the metagenomic data set. And these span 4 different phyla for these hosts. Again, we wanted to detect our viruses, so we used our same detection methods, and we were able to get 640 viral populations. We're able to link 11% of these viruses to hosts from these same soils, and more work is going on with this, so you'll definitely see something in the near future. So bringing it back to a heat map here, I now use colors to remind you this is no longer this cold, frigid environment, but this is this lush, fluctuating anoxic, oxic environment. So at the top here in black, we have our bulk metagenome samples. And then in blue and red, we have our Sept fractionated metagenomes. In the light blue, we have our 12C, our natural abundance. And in red, we have our enriched samples. So these are microbes that ate plant biomass. And these are the viruses that infect those microbes. So the first thing I want to point out is that by comparing the bulk to Sept fractionated metagenomes, we're able to get 8% more viral populations. So by doing Sept fractionation, we did decrease complexity and increase resolution on viruses. We're getting more viral diversity from doing the Sept fractionated metagenomes. This is great. Now, if we zoom in just on the active viruses, and the way we did this is that if they're active in the natural abundance and in the enriched samples, we took them out. So these are only ones that were recorded in the in the 13C enriched plant biomass samples. The first thing you should immediately see is that from the oxic samples, we have more diversity of viruses. We have more viral populations. So oxygen is definitely infecting or correlating with our viruses. The other thing you should note, though, is that we have unique viruses in our anoxic samples that are not in our oxic samples. So why is it that these viruses would infect hosts that have these slower metabolisms? Is this a niche differentiation, or what is this? So definitely going to be looking more into this. So to summarize and bring it all back together, from our first experiment, I first actually just want to say that the Sept process worked. We were able to identify active microbes and viruses, and this reduced the complexity, so we got increased resolution. And these are the metabolisms that we care about because they're actively interacting with the environment. So for our first site, this is the Bonanza Creek, this is our permafrost. Not only did we identify these microbes and viruses below freezing, so they are contributing in these frozen soils, but we also see this temporal succession. We see this change in viral community and the evidence that viruses can persist in the environment, but more work is needed to know in which way they persist. Switching over to our highly dynamic soils, we are able to link viruses to key microbes that break down and control the fate of this organic carbon in the soil environments. And that we saw that redox strongly influenced our virus activity, and that the Sept fractions recovered more viral populations. So to synthesize what we gained from both these environments is that we got a lot of cool novel viruses here, and the Sept fraction metagenomes helped us see stuff that we couldn't see otherwise. I also wanted to point out we put a lot more effort into the tropical soils with 8x more sequencing, but we only had about double the number of viral populations. And there can be many reasons for this. You could think maybe viral diversity doesn't track with microbial diversity, or it could be that there's less organisms reducing the metagenome complexity. Finally, the metabolic repertoire between the active host and the dormant host was completely different. So we need to think about this when we're looking at our metagenomes. Unless we're actually looking at the active ones, we can be seeing signals of stuff that's actually not going on. And how do we infer this if we're looking at biosignatures or if we're looking at gases and we're trying to relate it to microbes? We need to make sure that they're active when we're taking those samples. So I just want to acknowledge everyone at JGI and Lawrence Livermore National Lab that helped, especially my mentors, Steve Blazewicz and Jennifer Pepperidge. And with that, I'll take any questions.
Cool. Is that your little one? Some online.
You see that, Gary?
I'm just seeing it now. Let me see.
Method could be used for snow-dwelling viruses. What about aquatic viruses?
Yeah, so this technique can be applied in any environment. It's not just for soils. That's the one that I cared about. There has been work. I posted a paper earlier that uses Bondcat and uses nanosims to look at it with Bondcat's just another way to look at activity of in situ protein synthesis. I can also post a book from one of my mentors that she talks about how we can use stable Jennifer Pepperidge. She talks about how we can use stable isotopes to look at microbes and their viruses, and gives a great review on the different isotopes you can use, different compounds can be applied to, and what are outcomes and prospects of that.
Okay, Arvind has a question.
Hi, Gary. Thanks for the talk. I've got a quick question, and this is your observation that you're noticing in your, say, your soils in Puerto Rico. And because the soils go through all these variations, and I'm wondering whether the viruses you're finding in those ecosystems are more generalists, so that it's a mechanism for the virus to persist in that environment without actually leaving the gene pool. And so with that, you might have less diversity of viruses, which is kind of what I expect to see in these kind of systems, which is not far from a system that you would see in an agricultural setup where you have crop rotation or something, and you have reservoir species that the viruses go to when a crop is taken out of place, and then you go back into the same circle. So this is kind of what I think might be a reason you're seeing lower diversity compared to your other samples.
Yeah, that's actually really great. I took note of that. This is something I've been thinking about as well, which is why I'm excited to get more into the host linking. I'd like to know if there are these polyvalent viruses that can affect a broad range of microbes. So far, I've only done a nucleotide similarity via BLAST. Obviously, some more work with CRISPR and maybe another application to really see if I can get in on this.
Cool, thank you.
Thank you.
Here's another comment about doing a thought experiment as to how the SIPs might work in astrovirology or astrobiology context.
Yeah, so this is great. I mean, I always think of it more of a biosignature aspect of when we read one, how it can be different if a virus is infecting the host. But if we're going to take stable isotope probing to another planet to label something. I mean, that would be great. In terms of maybe we're setting a trap to look for life. We take something, it's labeled, we leave it, we see if anything interacts with it, and somehow have to control for abiotic factors. I don't know if Ken or Penny, if anyone else has any thoughts on that. I wish Kathy was here right now. She would be able to
she had
talk
to
about
step
that more.
out for another telecon. I think it's a really tough call about how to do that, but I think it's worth figuring it out.
Yeah.
And I think it's mostly a tough call right now because it hasn't received the same attention that, observing bacterial size things with their properties has been, had a lot of attention. So, I think this could be something to actually try to expand in a journal article. It's like, how could this be applied in that astrobiological context? So I think it's the next excellent question.
Gary, I just have some sort of technical questions.
Okay.
If I remember correctly, you didn't actually physically separate the virus particles, the virions, from your bacteria. This was just a whole metagenome, correct?
This is true. So sorry, let me specify that for now, because the way our pipelines work, we have to have a large amount of DNA in order to have the fractionation for the cesium-clair density, right? So currently, these are SIP metagenomes. I would definitely stay tuned for a potential SIP virome in the near future.
And sort of a follow-up on that, do you actually see bands in the cesium, or do you just fractionate and pull off what's the right size?
So I'm actually going to share the slides back with you. I had a backup slide in case there's a question. I make it seem easy, but it's not easy.
Yeah, I've done cesium too, so I'm kind of...
So I showed these clear, distinct bands, but that's actually not what you get. Really what it is that we get many bands, and this can be problematic because it's all about density. So if you have microbes that are high GC content, they're going to be more dense. You have less GC, they're going to be less dense. And if you put this in viruses too, because their genomes are small, this can have a larger impact. And then with microbes, you have such large genomes that you can imagine their genome is like spread out. So this is something that we're working on to try to fine tune. And we take many SIP fractions when we do the SIP fractionated metagenomes. Now that Kathy's back, I actually wanted to bring that question back up.
Oh.
There was a question about how would we use SIP if we were to send it on a rover into space and we were looking elsewhere? How would we be able to use like a stable isotope probing technique where something gets labeled and we can track it?
Well, I don't know if anyone's mentioned Viking and the labeled release experiment.
Cassie was going to mention that. Cassie Collins just mentioned that, although she didn't elaborate on it. Obviously, she nailed it.
One of my favorite missions was Viking and the labeled release experiment because it was really a life detection mission. Like that's what it was there to do, was to detect life. Inherently, it made a lot of assumptions of if there was life in the Martian regolith, the nutrient soup in which it would need to be metabolically active, just all these assumptions about life there having these sorts of requirements. So I think with a labeled release sort of experiment, you have to be very careful not to assume things about metabolic activity. So that is sort of the crux of that problem, and I don't know if I have a good answer, but I will definitely think about it.
We need to write proposals.
Well, I figure it needs to go in the paper after you guys figure it out.
Okay.
Okay?
Great.
All right, I'm going to add it. Yep.
And do you want to comment on cave viruses, Penny?
Yeah, I did online, but I pointed out that there's less than a handful of papers in English in the literature on the subject. We do know from being in the field at the Nika caves that are very, very hot and have no natural openings from just some very preliminary work that Curtis Suttle and his group did, that there's a huge load of viruses. And so that was something that we have used to actually indicate that the extraordinary microbial communities that we're finding there are indigenous, because where there are bugs, there are tiny bugs that are eating them, bugging them or something. And so, but no actual work on the identification of those was done in that case. I think that Dale Griffin has published maybe 1 to 2 papers in terms of cave virus stuff. This was work done with my friend and colleague Diana Northup at the University of New Mexico quite a few years ago now. And part of the reason, as I mentioned on the first day, that Barry was interested in a cave field trip for virologists was to actually stimulate interest in the field. And if people are interested in samples from caves, I go into many, many of them. So I think that one of the selling points for cave samples is that at least if it reflects in the virology what we see in the bacteriology and the studies of archaea, that because it's a geological environment that is highly partitioned physically with limited modes of transmission by way of air currents and fluids, that we see tremendous individual evolutionary experiments. And so the diversity is staggeringly huge amongst the bacterial and archaeal populations, and it's because we believe of this, partitioning. And so I would guess that we would see, an equal stunning diversity, not that viruses aren't stunningly diverse enough as it is, but, that would reflect that same isolation. So in a way, it's sort of an underground island biogeography situation where you've got all these isolated geological habitats. So if anybody is interested in cave samples and wants to actually do some serious work on them, you know where I can be found on email, I bet.
Can you repeat the paper again?
Which one?
It was the cave virus paper. Was there an actual paper that came out of it?
Yes. Dale Griffith was, or is it Griffin? Dale, he's at USGS in Florida. Is it Griffin?
I think it's Griffin.
Yeah, I think it's Griffin. Okay, so Dale. But anyway, he's at the USGS in St. Petersburg, Florida. And so he did some work with Diana looking at the viruses in parts, I believe it was Carlsbad Caverns. And he also has done other creative things about the, the microbial load coming across on dust across the Atlantic Ocean from the deserts of Africa and stuff. And a lot of that material gets deposited into caves as this material called terra rosa. And so that obviously carries all different kinds of life forms. Undoubtedly, there are viruses coming along with that as well. So maybe one of the things that I can do as a follow-up to this is assemble the feeble amount of literature that there exists in terms of subsurface viruses in that way, and make that available to the group. That will be a very short task.
Yeah.
Yeah, unfortunately.
I think that Felipe may have found it already, so.
Yeah. Okay, good. Thank
maybe.
you.
I don't know, that's human visitation, so maybe it's not. Oh,
yeah. I think that's the different one, but yeah, there are several.
Cool. Great.
And Laura G says she's actually beginning with some cave microbiology research in Mexico, or at least trying. So go, Laura. Or maybe if you're Mexican, it's Laura. Okay. I don't know. But, email me if you want to chat about it.
Okay.
So maybe we're ready for you, Ken?
I think I'm up. Yeah, I just need to move my window here. Sorry about this. It's little confusing.
Yeah.
It's always a problem to have 3 monitors. It's it's just one of these issues that one has to deal with. So okay, here we are. Come on, go and share. Which one shall I share? Let's try sharing this one. Okay, have you got my slides there? Good. Okay. I don't see anybody, waving or complaining here. So just wanted to finish up today. Again, thanks at the very first to absolutely everyone for participating. This has been absolutely fabulous. And I think we're going to get some really nice products out of it eventually as well. The background picture here is a place called Boiling Springs Lake, which is one of our main field sites. Probably a little easier to get to than some of the caves, but nonetheless, I think a really pretty fascinating place to look at. So what I wanted to do was, if I can change my slides here, there we go, is talk a little bit about some virus definitions. And we got some of this already with Evelyn right at the beginning, but just a bit of a way to sort of try and bring people again back together, and some people may have missed that talk at the beginning. So classical definitions of viruses were, I love this one, simply a piece of bad news wrapped up in a protein, Sir Peter Medawar. And that's how the vast majority of people think about viruses. And thanks to Nigel for pointing out that most viruses actually have a really pretty bad rap. And that's a lot of what I do, particularly in my public outreach, is that try and explain that they do have a very bad rap. And so if you ever go into the textbooks, you see this, they're very small infectious obligate intracellular parasites. Again, Evelyn did a great job pointing out that very small. All my students know if I put something in quotes and in red, I don't believe it. And so that gives you a bit of an indication what I thought about it. And then one of my favorite definitions, actually a lot like the one that Penny just used. A virus is a bag of nucleic acid. It's a very specialized bag, and it's usually very specialized nucleic acid, but that's basically it. But I don't like really any of these particular definitions. The one that I actually like the best has been going back into the literature, and Salvador Luria had this definition in 1978, which I really, really like, and we'll get back to in just a second what I really like about this. But viruses are entities, not saying anything particular about them, whose genomes are elements of nucleic acid that replicate inside living cells using the cellular synthetic machinery and causing the synthesis of specialized elements that can transfer the viral genome to other cells. And so that fits nicely with this overall image here. This is a lytic image, and you can look at the lysogenic images. You have an extracellular specialized element that interacts with a living cell. Then there's the nucleic acid that's inside this package that gets put inside the cell, that gets replicated and made into phage protein by cellular components. And so again, the key here is it's a cellular synthetic machinery. And one particular piece of that machinery, which is absolutely critical to all viruses that we know of, is the ribosome. No viruses that have been found to date have ribosomes in their genomes. They have things that modify ribosomes and modify translation, but none actually have ribosomes. And so that, I think, is a really nice way of defining the difference between cellular life and viral life. Then you get the assembly of these particles. Then these particles are released, and you go through the whole cycle again. You could also, of course, have replication of these infected cells where things will continue along. So that's again, I really like this definition. I think it's a very useful definition, and we can talk more about it later in terms of whether people particularly like it. But before I go there, I wanted to mention a particular piece of art, which is, I think, very relevant to thinking about viruses, and particularly in the discussion of whether viruses are alive or not. This, of course, is the famous surrealist painting by René Magritte, Ceci n'est pas un pipe. But as Patrick Forterre always loves to say, Ceci n'est pas un virus. And this particular virion, which is that entity which is built by viruses, is very different and very unique and very specific to viruses, but it's not the whole story. The whole story is really much more about the virus life cycle. And this gets us back to Joshua Weitz's presentation yesterday, where he's really talking about viruses and cells and how those viruses and cells come together. And once a cell has been infected by a virus, that cell, and particularly in a lytic case, but even if it's replicating in a lysogenic way, that's also really a virus. And so Patrick Forterre and Didier Raoul came up with this concept, which they called the ribocell, again, because these have ribosomes, and all viruses need cellular ribosomes to replicate, and the virus cell, which is where you have virus replication. So again, I love Joshua Weitz's presentation thinking about modeling, because I think you really need to think about viruses in a much more general case, rather than in this sort of, we're just going to look at these extracellular particles. And the extracellular particles, of course, are the virions, which we've heard about quite a bit before. And these are things that are really, really specific to viruses. Only viruses make these things. And they have these incredibly distinctive morphologies. Kathy was just talking about these, ways that you can detect them nicely with nanopores, particularly the tobacco mosaic virus, this long, stiff, rod-shaped form. Many of the bacterial viruses, this is just an environmental sample here, which have these heads and tail structures, and very often with icosahedral symmetry. And this kind of symmetry, again, these small dots that Evelyn was talking about before, I think this is really indicative of what a lot of people think about the viruses. And now she alluded to this as well, but my favorite kinds of viruses are the really, really weird virion-containing ones. And so these are a sampling of some of the archaeal viruses. Evelyn mentioned this Acidionus filamentous virus with these nano claws at the end. I'll talk a little bit more about our favorite viruses. These SSV1s that I've been looking at before have this spindle or like lemon shape. There's an individual virion right here. And then probably the most amazing of these were some virions discovered by David Prangishvili's group when he was in Regensburg, where they have these appropriately named Acidionus bottle-shaped virus. And these really do look kind of like champagne bottles that have usurped a birthday cake and put the candles at one end. How the heck you make one of these structures and what it would look like going through a nanopore, I would just love to know. But I think it's really important from an astrovirology point of view is to not get fixated on head and tail virion morphologies, but really start thinking about a lot of these other ones. And the nice example of that, and again, Evelyn brought this up, are the gyruses, the giant viruses. And the first time that I saw this picture, it just completely blew me away. Each of the little dots up here in the corner, each of these is a virion. And this is light microscopy. So light microscopy, you can actually see the individual virions. And this is an electron micrograph down here of one of these Mimi viruses as well. So virions are definitely not just small, they're not just head and tail, they're all kinds of different things. And even there are some viruses which have viruses that infect them. And this is the first case of the so-called virophage, the Sputniks, which infect the Mimi viruses. The other thing to mention about these giant viruses is that they have massive genomes. Mimi virus itself has about over 1 million base pair genome. And some of the larger viruses since then have gotten to almost tens of millions of base pair sized genomes, well larger than any bacteria, or not any bacteria, but many bacteria. But nonetheless, none of them to date have any of these ribosomal proteins in them. So massive amounts of diversity. We heard also from Evelyn that we've got these very different kinds of genomes that they could have. But we also heard from her that they're incredibly common, and we also saw exactly the same picture turned around a little bit in Simon's talk. If you look at seawater samples, lots and lots of little virions here, one poor little eukaryotic diatom, and a few overwhelmed bacteria and archaea. And I love this juxtaposition, I forget who first showed it to me, of the Hubble deep field, because I think these look incredibly similar to each other, but we may actually know more about these dots than we know about these dots here as well. So one of the things that you can do with samples like this, and as Simo mentioned, there's a lot of uncertainty in many of these numbers, you can do some calculations and you get to ridiculous numbers of virions on our planet. And Roger Hendricks kind of took some of these to heart and mentioned that this is what the Earth looks like with 10 to the 31 virions, most of them bacteriophages. And JBS Haldane said the creator must have had inordinate fondness for beetles. Well, if the creator really had as much fondness for beetles as he did for phage, the Earth would be this size. So clearly, the creator was much more fond of phage, as we've heard from many people already in the presentations, than she ever was with beetles. The other thing you can do, of course, is line all of these phage particles up end to end, and they end up, depending again on lots of orders of magnitude, of 2 times 10 to the 7th light years. Then these are just the virions on Earth. So that to me is sort of as astrobiological as this could possibly get. But that being said, one of the things about these very, very large numbers of viruses and their whole process, again, remember, this is not just virions, but viruses on our planet, means that they actually do play very important roles in some big biogeochemical cycles. We heard a little bit about that earlier, but I love this image. It's admittedly almost 15 years ago, a really nice review article by Curtis Suttle in Nature, just looking at what happens with the presence of viruses in oceans. And what they do, and again, we've heard about this in previous talks as well, is they recycle a lot of the dissolved organic carbon and keep it actually in the upper ocean, as opposed to having a lot of that disappear and this carbon actually sinking to the bottom of the ocean. So the presence of viruses is really important in some real on Earth, global biogeochemical cycles. And I think that one of the things, and we mentioned this in our astrovirology article, that people should be thinking about is thinking about these kinds of biogeochemical cycles on an early Earth, in some other astrobiological contexts. I think this is something that's really not been looked into at all, and something that I really would hope that we can get people convinced to start to look at. But it's not just in the oceans. It turns out if you look at our genome, it's also massive numbers of viruses. Sorry, the animation here got lost in the process. This is a linear representation of our human genome here. This gray part here is really clearly viral derived, retroviral-like elements. About 8% of our human genome are these retroviral-like elements. Line elements and sign elements, people can argue about whether retroviruses came from these retro-like elements. Some people argue one way, some people argue the other. But be that as it may, they're over 40% of our genome, compared to our protein coding parts of the genome, which are 1.5%. So basically, we're more viral than we are human. And that also is something that I think most people don't think too much about. They think viruses are bad. Well, then we're bad too. We can argue about that as well. But I think that we've also gotten some really nice examples of some of the chats we were talking about earlier, talking about good viruses and how viruses can be good. My favorite example of good viruses is the set of viruses, the thermal tolerance viruses that have been studied by Marilyn Rusink and some of her colleagues. This is a nice review in 2011. Also, I think we got it referenced later. This is a plant, in fact, something called Dicanthelion lanuginosum, also known as panic grass. We actually see this around all of our thermal sites when we're going and sampling for the extremophilic viruses. This panic grass basically tells you where not to step because it's at 50 degrees plus. But it turns out that 50 degrees Celsius, the plant can only survive under these conditions if it's infected with a fungus, and that fungus is infected with one of these thermal tolerance viruses. So this, I think, is probably the best example of a good virus, because without this good virus, this plant could definitely not survive. And there's some great, there's a science article, in fact, that Marilyn wrote about this. You can clearly see that the plant does not do well at 50 degrees unless it has both the virus and the fungus. So the last thing I wanted to talk about in terms of a general virology point of view is talk a little about the origin of viruses. And I know this is highly controversial, but I just wanted to throw it out there. Mark Krupovic, Valerian Dolja, and Eugene Koonin came out with an article in Nature Reviews Microbiology, just came out a little bit earlier this year, thinking about the origin of viruses and whether we're ever going to know about the origin of viruses or where they came from. Was there a universal viral common ancestor? I think these are very open questions, but at least the way that Krupovic, Dolja, and Koonin put it, they thought that there were these autonomous replicators which were probably present at a pre-cellular stage of evolution. And you can see some of that by looking at the phylogeny of some of these genes here, and I'll let you read the paper, take a look at it. One of the big questions that they tried to answer is, how do you get capsids? How do you get virions? And that seems to have been potentially, for the very first viruses, they picked up virions from some parts of a primitive cell. But clearly, over evolutionary time, there have been massive amounts of gene transfer that have allowed the development of all of these other friendly or not so friendly viruses that we know today. So that's the background stuff. I wanted to spend a couple of minutes talking about some of the stuff that my lab is doing. Can anybody read this? So I've been told the translation is hell virus. This is Chinoike Jigoku, Beppu, Japan, where I was fortunate enough to go before. And we're pretty sure, but not absolutely certain, that this is the place that our favorite virion comes from, SSV1, which we've solved the structure of together with Mark Murray at the UT Medical Branch. And this background here is actually Boiling Springs Lake. So we've been studying the structure of these viruses. We've been studying the genetics of these viruses. Most of this work was the work done by Eric Iverson. And one of the things that we found there is that this virus genome is incredibly tolerant to mutation. We can actually mutate about half the genes in this viral genome, and the virus is still okay, which is very bizarre. Most viral genomes, you start messing around with some of the virus genes, they become unhappy very, very quickly. And again, this is all the work of Eric Iverson. Here we are in Yellowstone, standing next to what we thought was the hot spring that Taq polymerase came from. Actually, it's over here about 1/4 mile. So we're close. So why are we studying these viruses in the hot springs? Well, one of the reasons is they're just bizarre and interesting because they've got these really fascinating structures. And certainly NASA is interested in viruses in extreme environments, just thinking about extreme environments and what this means for understanding viruses in general. And we also got a microbial observatory project funded by the NSF for this place called Boiling Springs Lake in Lassen Volcanic National Park. And this sort of defines extreme environment. High temperature, 95 degrees Celsius, a pH of 2, relatively easy to get to. Low temperature is 50 degrees. And so we actually were able to detect some eukaryotes in this system as well as bacteria and archaea. And we, of course, were really interested in the viruses. And none of my students wanted to go out on the lake and collect samples. So we had this little ROV to go out on the lake and collect some samples for us. We tried to find viruses here. We had no luck whatsoever. So we actually did a metagenome, and we've heard lots about metagenomes already. One of the things about metagenomes is you usually lots of material. And so these are a couple of my graduate students, again, Eric Iverson, and then the leader of this set of work, which was Jeff Deemer, collecting samples, bringing them out, and doing some metagenomes. And to make a long story short, in this particular metagenome, we found this very, very strange viral genome that looked like a recombination between a single-stranded DNA virus and a single-stranded RNA virus. And this is a recombination that was never supposed to happen. Everything that people had said, it doesn't happen, but as we heard from one of the previous talks, viruses don't care. And I think that was Evelyn as well. Viruses don't care what we call them, they just do it anyway. And so somehow a single-stranded DNA virus and a single-stranded RNA virus got together and generated this, what we're calling now a crucivirus. And stay tuned, Arvind and my lab will be publishing a really interesting paper on this that will kind of put some of his recombination machineries to shame. At least we think that might be the case. So finishing up really quickly, I wanted to talk about virus biosignatures. This is a sampling site in Yellowstone National Park. You can tell the hair is a little less gray, so this is a few years ago now. Here I discovered a virus that we called Sulfolobus turritidicosahedral virus when I was a postdoc in Mark Young's lab. Liang Tang and Jack Johnson's lab solved the structure of this absolutely beautiful particle. We got really excited about these projections at the five-fold axes of symmetry, but they noticed something I think even more interesting, and that is that the coat protein of this virus, which is now infecting archaea, was practically identical between archaea, bacteria, and eukaryotic cells. Is this horizontal gene transfer? Maybe, but that would be between archaea, bacteria, and eukarya. We think this is unlikely. Could be convergent evolution. We think it's an example for a common ancestor, but a common ancestor that now would be around since you had the distribution of bacteria, archaea, and eukarya. So overview of this is the papers. Basically, if you look at one of these cellular trees of life, eukaryotes here, archaea here, and bacteria here, now we think that there are some of these structures, again, that might provide some biomarkers that are really potentially extremely ancient. Whether that primordial virus back here looked like this one, probably a little bit open to question. But this is all clearly extremely indirect. And so this is where I think one of the people was chatting about, virus taxidermy as well as virus taxonomy. We got interested in viruses in the rock record. And this is where I have to thank one of Penny's, predecessors for funding this absolutely crazy project on virus fossils. So we got some Director's Discretionary Fund money for this from Carl Pilcher, and basically went looking for virus fossils in some of these hot spring environments. And when I say we, most of that is Dr. Jim Laidler, who was an anesthesiologist before he came to work in my lab and got a PhD. Nice to have all of his anesthesia equipment when you're out in the field. But to make a long story short, what Jim found was you could take virions, particularly those like T4, and silicify them, i.e. coat them in silica, which is not fossilization, as the geologist will tell me. It's mineralization of various different viruses. We mineralized tobacco mosaic virus. We mineralized bacteriophage T4. We also mineralized some of our hot spring viruses. And what those look like for short periods of time, literally hours, here's some silica treated and here's some non-treated bacteriophage T4, that you got some silicification happening, but unfortunately over time periods more than literally a couple of days, you lost any kind of morphological signal, at least at the resolution that we were able to look at here. But then serendipity raised its, I think, beautiful head in this particular case. And Jim took a look at what actually happened to these viruses when they were silica treated and when they're silica coated for a short period of time. And what he found is that some viruses didn't lose their infectivity at all in silica treatment. Some lost a lot of infectivity, which is here on the y-axis, and some were kind of intermediate. Hot spring viruses lost less infectivity than bacteriophage T4, and some human viruses, this is vaccinia virus, actually lost a huge amount of activity. And this is kind of what you'd expect once you get a silica coating on things. Our big surprise came when these viruses, when we put them in conditions where there wasn't silica, regained their infectivity. So we have what we're calling the zombie viruses here, which have then come back to life based on this silicification and desilicification. And a number of people were really excited about this. We got viruses, we're gonna have viruses on meteorites, they're gonna be transferring, we're gonna get all the Martian nasty viruses being transferred to this planet. Turns out that after about a month, these lose their infectivity completely. So unlikely that this process, at the very least, will be able to preserve something via spaceflight, but endolithic viruses, maybe. It's certainly a possibility. So with that, I just wanted to finish up with some of our proposals that we had in our astrobiology article. And basically, I'm just going to leave these up here and answer questions on the rest of my talk. And then hopefully we can come back and revisit some of these questions a little bit later on. So with that, I'm only 5 minutes over, which as an organizer, I think is pretty good. And I'll take questions if anyone has any at this point. Turn the lights off so maybe you can actually see me. Nobody wants to ask questions?
I have a question. It's probably pretty naive, but is there any way that you could imagine kind of creating that zombie virus other than mineralization, or maybe a different type of mineralization where they could be preserved for longer durations of time, and then their morphological features might not be preserved, but that they could be brought back after an extended period of time?
So that's a great question. We tried with silica because silica is relatively straightforward. There has been some work of Jennifer Kyle looking at iron and seeing if virus interactions with iron could help to preserve them, but nothing really that seems to be able to stabilize them any more than silica has been able to. So I think that that's we've probably done about as good as we can with these particular viruses, and mostly we've been looking at bacteriophage T4. I have much higher hopes for things that you might find some viruses in endoliths. Maybe some of Gary's soil viruses might have some kind of way that we can look at those. I really don't know, but I think it's a wide open area for people to start looking at.
Yeah, one of the things I wanted to mention just as a follow-on to that, Ken, is that microbial communities in general self-fossilize in the highly mineralized environments of caves. And so a lot of that is, excuse me, because there are chemolithotrophic organisms there that are getting their energy out of transforming one mineral into another, typically from an oxidation state to a higher oxidation state. And because of that, they're essentially coating themselves in minerals. And then they're also undisturbed by weather. So it's a wonderful preservation environment. And I think, not to keep harping on caves, as wonderful as they are, but I think, it's one of those places where one might be able to actually find undisturbed in situ viral fossil materials, and caves come in all different kinds of geochemical flavors. So sometimes there's silica gel that is fossilizing things. Very often it's carbonate, sometimes it's gypsum, it can be copper, it can be iron, it can be sulfur. So there are a lot of possibilities for actually looking. If we knew what the heck we were looking for, we might be able to point virology folks in the right direction or provide samples. Just saying.
Yeah, no, and I completely agree. I do think that this is something that we should be looking for, and something that we should definitely put into a review article and or white papers. I think we need to look at some of these things. And part of the problem that we had with Jim's study and the original silica coating of T4 was actually a lack of resolution. It wasn't a case of really, the morphology was gone, but if we had some kind of higher resolution way of looking at chemical structures, we probably would have a really nice biosignature. It's just that I don't know enough about that kind of technology, and I'd love to talk to more of the engineers who are trying to think about some of these things.
Right. Great. Thanks.
Has anyone subjected them to like a space environment like the tardigrades to see if they would be okay after a while?
So that's another great question, and I think Penny sort of alluded to this when she was mentioning, numbers of papers in English. There are a couple of Russian papers actually. So some of the very early Russian space program looked at some viruses, including tobacco mosaic virus, in the space environment, and they seem to do reasonably well, and they didn't lose too much in the way of infectivity. But these were all really pretty short-term exposure kinds of experiments, and so. The longer-term exposure experiments, we were hoping that with our silica treatment we would get something that we could try. But if we lose infectivity in a month with desiccation at room temperature, highly unlikely that's going to be something which is going to survive in a space environment. We could try, but I think that we need some more, maybe more simulation environment before we actually go into a space environment.
Yeah.
Fair enough.
There are some other comments here.
Yeah, somehow I've lost my comment window here.
People are asking about ancient folds on viral proteins. I'm not sure that whether you can answer that or not, but.
Okay, so let me see. I'll elaborate more on the ancient folds on viral proteins. So SH3 or OB folds. Okay. Yeah. So sorry about that. Again, trying to deal with too many screens here simultaneously. But yeah, so the fold that I didn't have a chance to get into, but the folds that I mentioned for our viruses, the ones that, again, the STIV that we find in Yellowstone Hot Springs and also has a similar structure to the bacterial virus, and the ones that you find in some eukaryotic viruses as well, not just adenovirus, but a number of different ones. These are what are called double jelly roll structures. And a lot of work has been done on this with Mark Krupovic, Eugene Koonin, but also Dennis Banford, some of the structural biologists. And so this double jelly roll and also single jelly roll kinds of proteins are very well conserved in terms of their structures, but not very well conserved in terms of their sequences. And so what I think would be really interesting to look at, again, potentially from a biosignature point of view, is if we can get to that kind of resolution, can we find double jelly roll kinds of proteins anywhere? And so I think that's an open question, but certainly there's a lot of these double jelly roll proteins, but there's also something called the HK97 fold, which is a very different kind of protein structure that's also found in bacterial viruses and eukaryotic viruses, not in archaeal viruses yet, but archaeal viruses are incredibly understudied. So it's distinctly possible that that kind of structure is actually there.
Let's see.
Okay, there's one about the examples of the Russian papers on viruses in their space program that I mentioned. Those are referenced in our review article. I could go and dig them up, but if you go into our astrobiology article, and if you don't have a copy of it, just let me know. I can send it to you. So those are the two that I was able to find. There may be some other ones. There may be other space literature that hasn't been published, but at least those are the ones that I'm able to find. But they were from the, I think, 60s and 70s, most of them.
Okay. There's also a comment by Cassie. She's saying to suggest to collect viruses from places that normally spend most of the time desiccated. So I guess arid environments to have a better chance of long-term preservation.
Ooh, hunt for the desiccation virus.
Yeah. Sounds like an expedition to me.
Yeah, I think people have looked in the Atacama. I'm not absolutely certain.
Yeah.
I think, Arvind, did you have a question you wanted to ask?
You're muted, Arvind, so if you want to talk, you need to unmute yourself.
Maybe not.
Okay. No, he said yes, but maybe you'll fake. Marco, can everybody unmute themselves now?
Yes, they can.
Right. Ken, I had a quick question, and that was to do with desiccating the viruses. And I'm wondering, because in those kind of scenarios, you're deriving the water away, and that destabilizes nucleic acid. So I'm wondering if anyone has done any kind of analysis at a sequence level to see, are these viruses more prone to deamination, any other kind of base changes, coupled with when the viruses do recover, are you seeing variants or mutations that are at a higher rate? So what I'm trying to get to is, are we in these kind of cycles where we might be getting into, say, the environments where they go through these rapid changes of environments from wet climatic conditions to dry to wet again? Are we seeing an accelerated boom burst period of virus evolution at any given period of time?
So, great question, Arvind. The short answer is we don't know. The longer answer is actually one that ties into someone else I think who asked on the chat room also about basically what happens to the viruses when we desiccate them and when we do the coatings. And as you know, this is actually really critical to my side hustle, which is trying to use this technology to preserve vaccines to get them out into the developing world. So if coating and drying does something to the viral genome, this could clearly be a big problem. So it's something that we definitely need to look at.
Oh, thank you.
There's a question from Ishmael somewhere here.
Yeah, about isotope fractionation by AMGs.
Yes, of AMGs, yeah.
So wonderful question. I'm by no stretch of the imagination an expert in looking at isotope fractionation, but I certainly have looked at and read papers of people who've looked at fractionation, particularly of C13 in rocks and in microbes. And what they see is that depending on the particular kind of metabolism that's being used, there'll be more or less enrichment, in this case particularly of C13. If I remember correctly, methanogens enrich a lot more for C12 than C13, and Kathy may know more about this, Penny probably knows more about this than I do. But I don't think that anybody's looked at any of these auxiliary metabolic genes and whether there's any difference in terms of fractionation. And the reason we put this into our paper, and you may remember from that last slide that I had up before, is that this might be some kind of virus biosignature, or a biosignature that there had been a virus infection at some point in the past that could be detectable in the rock record. I hope that answers your question.
I'd like to add, maybe just hypothesizing, thinking about this, that when a virus infects its host, it wants its host to be efficient, it wants to do its duty. So you could say if it's going to be increasing respiration or any type of metabolic outputs, it's going to make it more efficient. And by more efficient, does that mean that's more likely to select, if we're going to use carbon 12C versus 13C, or because it wants to push things out so quickly, it's going to discriminate less, and there's actually going to be an enrichment of 13C in that isotope, of that isotope.
Sounds like we need to do experiments.
Yeah.
Or write proposals to get funding to do experiments.
Yeah.
I mean, we can, if we look at anything, like I studied nitrous oxide before.
Yeah.
For biotic, the fractionation factor can change, and it can change by who's doing it. Even within the organism that's doing it, you can look at various ones, and there's this fluctuation. And I never really understood this, but I definitely think it has viral influence.
Yeah, and I think that just the fact that those auxiliary metabolic genes is a pretty new thing that people are thinking about, and it's great that Nigel talked about it earlier as well. And certainly Simon has been very, very involved in looking at some of these things as well. So I think this is a very much a expanding field, and hopefully something that people are going to start to look at in more detail.
I've got something else to add, which I think might be very important, and this is kind of looking at environments. And one of the things is that people have been looking at, obviously, climate change experiments and impacts on viruses as a consequence of elevated CO2 levels or temperature.
Right.
And there's very little been done on it, but there are 1 or 2 papers that have been done in the context within the plant industry. And they've identified that elevating the CO2 level from, say, where we are right now, roughly 400 plus to about 700, what we do get is 3-fold increase in the rate of replication of viruses. So the viral load within a host increases by 3-fold.
Wow.
And this is in the case of barley yellow leaf co-virus. At least this is something that we know with some viruses, but this might be something else to think about in terms of environmental factors that could govern replication rates, turnover times, and exploration of sequence space in terms of adaptation.
Yeah, it's a great idea. I think it's something definitely to explore. Okay, I think this is also the time in the schedule where I'm supposed to be talking about what's next. Don't want to. I know people need to leave here, and it's getting late for people in other time zones. So, Penny, did you want to say another couple of words?
Just thanks to everybody for attending. We've had a huge participation, and so we're delighted about that. And I can't wait to carry this forward into watching everybody shape this into a paper. I think there are many revelations to me as a microbiologist focusing on cellular organisms. Lots of jargon I didn't understand, but I'm getting up to speed. And I think there are a lot of subtle evolutionary concepts that are emerging from the viral field, as I understand what you guys have been talking about for the last 2 days, that are different in a lot of ways from the way we think about things with respect to bacteria and archaea and even protists and whatnot. So I think coming together of the minds across this divide is really very useful, and getting these ideas into the astrobiological context out into the literature will not only help the astrovirology case, that this is a valid way to spend some of NASA's money in the future for funding some of this stuff, but also I think it's a way to get some of these ideas into the other part of microbiology that is thinking about things in a different way, and perhaps vice versa. So I'm seeing the potential for cross-fertilization. So I want to thank everybody. I want to thank all of you guys who scrambled around to get this organized, Gary and UCAN and Kathy and our IT people, Marco and Greg. And with that, let me turn it back to you for last words. And what's next?
I guess the last words are, and I think there's something Gary mentioned as well, is whoever is interested in contributing to review articles, white papers, etc., please get in touch with us. At least with me, email is the best way. I've seen some people, ping me on Twitter. The problem with Twitter is it's the fire hose approach, and so please send me something. Either direct message is great, but email is by far and away the best way to reach me, and probably best to send to Gary and or Kathy as well. And we will be in touch again, probably early next week, sometime, to at least sort of put together an email list, think about strategizing for these next steps. Gary, do you have anything?
I also just wanted to say, if anyone has feedback, please give it to us, either email or here in the chat. It would just be great to know if this was a success in your opinion. Any ways you think we can improve, we are looking to definitely improve. Anything you thought that was missing that we could help with an understanding, we will keep the website live and it has resources available there. If you have any other question, we're happy to add stuff as well to help push this field.
Yes,
and
please.
Also, we have Kathy here as a resource in terms of life detection, and we're very interested in the translation of these science ideas into what can fly on future missions. And it looks like we have, a robust future in life detection missions to a number of different bodies. Certainly some way to actually look for viruses and identify them in some fashion, or virus-like things, or what are they called, mobile elements, or I don't care what you call them, doodads is what I said on the chat. We're very interested in figuring out how to do that. And all of these things take a very long time to develop and figure out, right?
Yes.
We still are really very much in the infancy of actual life detection rather than just looking for bags of chemicals, which is, part of the approach, but we need more. And so folding in these other life forms, as I think of them, they're true life forms in my view, I think is really very important, and now is the time to really get on board with that. So ideas for instruments, ideas for approaches,
Techniques that can translate into instruments.
Techniques that exist that haven't been used with viruses before, or that are used on Earth but are not part of our pantheon of instrumentation that we're already using on space missions. So all of that is very fluid, and we're dying to hear about that. So contact Kathy, me, both of us, whatever, any combination of us, and we will, get back to you.
Yes, maybe not immediately, but we will get back to you.
Oh, no, not this afternoon.
No.
Were there any last minute questions that anyone wanted to ask? It can be on anything.
Okay, my final parting comment, viruses rock.
Viruses can be in rock. That's what I hope.
That too.
Thanks, everybody.
Thank you so much, everybody. Wonderful.
Enjoy the rest of your evening or day or morning or whatever time
Wherever
it is.
you happen to be.
Thank you, Marco. Thank you, Greg.
Thank you.
Thank you for the NASA technical staff that helped out.
Yep.
Yeah.
Flawless execution, as several of the comments are. So.
Yep.
Fantastic.
Super.
Loved it. Thanks,
Bye,
everybody.
everyone.
Bye, Ken.